Good morning. Today is Monday, November 15th, and we are, believe it or not, into our fifth and final section of 430. So we are rapidly approaching the end. There's a lot of material to cover in here in a short period of time, so it will be a busy section. And one of the challenges to this section is, one of the things we've emphasized for the past two units have been when you look at a bone and you look at that nice big round head of the femur, it kind of shows you what it's going to do. We actually drew the origins and the insertions of muscles, knowing that a muscle is going to pull from the insertion towards the origin. So by looking at it, we were able to know what it was going to do. Is the part of your brain that moves your foot, does it look like your foot? Does the part of the brain that allows you to see look like a big eyeball?
No.
No, of course not. So one of the challenges is this one is going to be a little less intuitive. We're going to have to learn the parts of the brain that do certain things, and they don't give us any clue by looking at it. So that is going to be one of the challenges we're going to deal with in learning the material. Although there's some tricks we'll use to help us with that. And so we'll see that as we work our way through that. There is also a fair amount of homework coming up. We've got a lab stare, we've got a physio-X, we've got a histology review that will be graded for correctness. This was not done to be punitive. It's just the way that it fell out. And let me make sure I have this right. Yep, absolutely. Again, the 24th, I know, is the Wednesday before Thanksgiving, and that is a day that is challenging for a lot of people. I know my kids have that whole week off. They're teenagers and they don't want anything to do with me anyway. So it's not as big of a deal for me, but I know those with younger kids, it can be an issue. And then there's holiday travel and everything along those lines. However, there is going to be a daily quiz on that day. I'm warning you about that now so that you can make that decision based on that. And again, it's 5 points. Hopefully at this point in the class, those 5 points won't make or break your grade. However, the way the class material fell out, we will be discussing our cranial nerves. This is not an assignment. This is something we will be doing in class. We'll be going over the cranial nerves together, kind of in a similar way we went over the muscles together. Depending on how this section works, and I will tell you right now that we tend to always run long in this section. There's always way more material than we have a chance to cover, so we fall behind very, very easily. So I can tell you right now, I don't know how much sensory stuff we are going to get to. And if we don't get to sensory stuff, these cranial nerves could be as much as 25% of your, let's say it this way, of your lab exam. So we're going to be going over that together in class, and it is a huge amount of information. So again, I strongly, strongly encourage you to be here in class on Wednesday. So I wanted to, again, because this was not done to force you to try to be here on Wednesday, it's just the way the schedule fell out. So I'm making you aware of these things ahead of time. So hopefully you can plan accordingly to be there. All right, one last point. Again, as you know, the physio-X is not necessarily hard, but it can take a fair amount of time. And this physio-X that's due on the 29th has 9 activities. So as you know, this is probably not something you want to wait till Sunday night at 11 to start working on, because you're not going to get a whole lot of sleep if that is the case. All right, so that is the game plan. That is what we're going to be looking at for the next couple of days. Today, we're going to start with our nervous anatomy and our introduction to the nervous system. And then we'll move on to physiology from there, and then our brain anatomy and histology in lab. All right, questions on any of that? All right, excellent. Let's go ahead and dive into lecture then. What is the function of our nervous system? What is the function of nervous tissue? This is the part where I say things out loud and you guys answer out loud, or at least type in it on the chat screen.
Communication.
Yeah, there you go. All right, remember one of the things we talked about is how the nervous system is specialized for communication. And of course, as we now know, that nervous system, along with the endocrine system, uses that communication to help to maintain our health and our homeostasis. And how is it that the nervous system communicates?
Neurons.
True, and what do those neurons do? We learned about this in our muscle system.
Very action potential.
Yeah. And as we learned, action potentials are big, positive electrical signals. And I don't think we necessarily said this per se, but what else can we say about these big positive electrical signals? What's another way we could describe an action potential? It's big, it's a positive electrical signal. What makes the nervous system different from the endocrine system? Well, let's think about this. Both are specialized for communication. So if I put my hand on a hot stovetop, do I want to wait till those damaged cells have a chance to make a chemical signal to release it into the interstitial fluid, where it can go into the blood and circulate throughout the body to finally get to my brain to tell me that there's a problem? And it can make a hormone and release that into the interstitial fluid that's going to get in the blood, and eventually we'll get back to the hand to tell it to move.
We have sensory, something like adapters.
It's like reflexes.
Right. True. It is an example of a reflex. Absolutely. But the key with the electrical signal is electrical signals are fast, right? It's a big, positive, fast electrical signal. As you guys mentioned with that reflex, when I touch something hot, a fast, rapid, big electrical signal goes to my spinal cord. From my spinal cord makes a decision, sends that signal back to my hand, and I pull it away before I'm even aware of the fact that I should be cursing. Right. So the advantage of this nervous system, really the big difference between the nervous system and the endocrine system from a communication standpoint, is their speed. Right. There are obviously other differences as well, but a big one is speed. So we have a big, fast, positive electrical signal, that action potential, that allows for rapid communication. Now, of course, the nervous system is also responsible for a ton of other stuff as well. What else does our nervous system do besides maintaining homeostasis? Some of it is being done right now. Coordinates our movements. Excellent. What else?
Is it digestion?
True. Controls our digestive system. Absolutely. Produces speech. Allows you to hear speech or see me on camera waving to you. Absolutely. And can you just experience things that are happening right now?
You can like remember things and memories.
Memory, emotions, absolutely. All of these things and more. Our perception of the world around us, our behaviors, our movements, our memories, speech, all of these are things and more that our nervous system is responsible for, and our nervous system does for us. So it is a huge, big, powerful system. Absolutely. And of course, the study of the nervous system is the field of neurology, or the field of neuroscience. All right. Actually, let's do this on the whiteboard. Just like most things we have talked about inside of this class, we can identify the functional components of the nervous system. And we can identify the structural components. Functional components are what the nervous system actually does, and there are 3 functional divisions to those. And let's make these, we'll make them blue. 3 divisions of the nervous system. The first is our sensory division of the nervous system. And what does our sensory division of the nervous system do?
It receives information like inside and outside of the body.
Excellent, I
On the
like
side.
that. Excellent. So our sensory, try that again. Our sensory, wow, I did that way wrong. Our sensory division receives information from inside and outside of the body. Excellent. So that is our sensory division. Its job is to then send that information to our second big division. And this is that command center we've been thinking of from way back when we started talking about homeostasis. But in this case, we'll call it the integrative division. And this integrative division is responsible for several things. It receives the information from the sensory. As we know, it processes that information. It also compares that information to what we know or to our memory. And then once it compares that information to our knowledge, to our memory, it then makes a decision. And once it makes that decision, it then sends information out, and it sends information out to what we call the motor division. And what is the job of the motor division?
To affect a change in the body.
Yeah, its job is to affect the needed change in the body. Absolutely. And of course, as we learned when we were talking about homeostasis, our integrative system sends out information via this motor vision to our effectors. And what are the things that we said could be effectors in the body?
Skeletal.
Right, yeah, absolutely. Axonal movement, so that would be our skeletal
And
muscle.
then cardiac.
But we can't forget about cardiac muscle.
Smooth.
Smooth muscle. So all of our muscle flavors. Anything other than muscle that can be an effector?
Gland.
Yeah, and glands. Excellent. So we have skeletal muscle, cardiac muscle, smooth muscle, and glands. So these are the 3 main functional divisions of the nervous system. Sensory. You see a bear with an axe. That information is then sent in to our integrative center, where we process that information and compare it to our knowledge. Bear, bad. Axe, bad. Therefore, bear with axe, very bad. And we make a decision to either fight that bear with an axe or to run away quickly. And either way, we are then going to send that information out to those effectors so that our heart can beat faster, our pupils can dilate, we can run, we can do all those things that we need to do to get away with that from that bear with an axe. All right. So again, these are our 3 main divisions of our nervous system. And again, I've got these pretty pictures here. Your book does the same thing. We have our sensory function, we have our integrative function, and our motor function. And they use a slightly more benign example. You see that glass of water, you process the information, you haven't had any drink for a while, so you control the motor system to go grab a drink. All right. Questions on those? All right, excellent. Now notice, and I'll go back to our picture to add to this. Notice that when we talk about our functional components or functional divisions of the nervous system, notice they differ in the direction they carry information. Notice our sensory information goes into the central nervous system, or goes into the integrative system. And we have a fancy term for something when it goes into. We say that this is an afferent pathway. Whereas coming out of the integrative center, our motor division, let's cheat, make this fit. And so what do you think the fancy word for that is?
Efferent.
Exactly, it's an efferent. Afferent means going into, efferent means coming out of. So this is an efferent pathway.
On the lecture slides with the example from our book, it says input and output. Would you prefer us to use the afferent and efferent pathways instead of input and output?
So great question. It is not incorrect to say that it is input coming in and output going out, right? And again, to use kind of more computerized type terms, there's nothing wrong with that, that the sensory information is the stuff coming in. However, when we talk about the direction, these pathways, and because we'll actually be talking about the physical and describing some of the physical components that make up these pathways, we don't want to forget about these terms afferent and efferent.
Okay.
We want to
Thank
definitely
you.
understand what those mean. All righty. So those are, again, functions, but as we've also mentioned, we need to be able to talk about structure. And we haven't talked about our nervous tissue yet. We've talked about there's the 4 tissue types in the body: epithelial tissue and connective tissues, which we learned together for an exam. We just finished learning about muscle tissue. And so now we have to talk about nervous tissue. Like all tissues, nervous tissue is made up of cells. And there are primarily 2 types of cells, 2 big general types, let's say it this way. There are thousands of specific types of cells, but 2 big general types of cells that we find in our nervous system. And those are the neurons and the glial cells. Now, neurons that we've kind of already, let's actually do this. Neurons we've kind of already talked about. These are the ones that are the functional. And as we know, these are the ones that produce the action potential. And the glial cells are basically cells that assist the neurons, help to maintain conditions, to maintain the environment so that the neurons can function properly. And these cells make up the tissues that make up the structures of our nervous system. And of course, the most obvious structure is the brain. So let's go ahead and go back to our whiteboard to figure this out. Here we have our structural components, the components of our brain, and we'll do this in red. Brain obviously is one of the big structures of our nervous system. What's another structure of our nervous system?
Spinal cord.
Spinal cord, excellent. Perfect. But there are other structures as well, like what? All right, excellent. Augustina, you are correct. If you remember, and let's cheat a little bit. I'm going to move these down. We've learned a little something about those neurons. We've learned that the neurons, I'll do this in dark red. As we've seen, a typical neuron has a centrally located cell body with those dendrites that we learned about and a big long axon. So we have axons and cell bodies. And so you are correct. Some of the structures of our nervous system are just made up of cell bodies and just made up of axons. Our brain and our spinal cord have both in them. But there are other structures as well. So if, for instance, as Augustina pointed out, we have a bundle of axons. Do you have any idea what we call a bundle of axons? Okay, no, you know it.
Gray matter?
Say again?
I don't know. I feel like we did this in our review or pre-lab. I just don't
You may
remember.
have, absolutely. A bundle of axons. My children get on mine quite a bit.
Network?
True, no, though I wouldn't say that my children get on my network. That, although, well, that is true too. They do get on my network to use there. But what else do my children, even without their phones, get on? There we go. Sarah's got it. Faustina, nerves. Nerves are a bundle of axons. All right. Now, nerves, as it turns out, comes in two flavors. There are cranial nerves. And actually, you know what? I'm going to change the size of this. What do you think a cranial nerve is? Well, it's a bundle of axons that does what? All right, well, maybe it'll help if I give you the other ones. They're also spinal nerves.
It's a location.
Yeah. What do you think the difference between a spinal nerve and a cranial nerve is?
One's in the head and one's in the spine.
Closer, absolutely. Although let's be a little bit more specific. You got the right idea. Cranial nerves come off the brain. Spinal nerves are bundles of axons that come off what?
Sign.
Yeah, the spinal cord. Absolutely. Just out of curiosity, any idea how many cranial nerves you have?
12 pairs.
12 pairs, absolutely. And you were going to learn everything you ever wanted to know more about those 12 pairs of cranial nerves. Any idea how many spinal nerves you have?
31 pairs.
31 pairs, excellent. The good news is those are all pretty much the same, so we're not going to have to worry too much about that. Excellent. So let's see here. Let's move this up here. That right there. Any idea of another structure?
Don't we have sensory, like special sensory
Excellent.
structures?
We're going to have special sensory structures. Or receptors, let's say it that way. Like the retina of your eyeball that allows you to see light, like the special hair cells in your ears that let you hear. We've already learned about some of the specialized cells, specialized receptors in the skin. For instance, those pressure receptors or the tactile receptors or all of those types of things. Excellent. We have a bundle of axons. Do you think there's such of a thing as a bundle, a collection of cell bodies? Meninges are actually protective structures. So that is something we are going to talk about, but they're really just the bubble wrap and crepe paper we put on the outer surface to protect the brain. So it isn't so much functional as it is protective wrapping, kind of like our serous membranes, right? The peritoneum and the pericardium and those things that we talked about. Well, we do have bundles of axons, but we can also have bundles of cell bodies. And these bundles of cell bodies are known as ganglion. Ganglion being singular, ganglia being the plural. So just write that out. So ganglia is more plural, and ganglion is the singular. So those are bundles of cell bodies. So we have our nerves, we have our cell bodies, we have our ganglia. The other thing we have are these collections of nerves and ganglia. These collections of nerves and ganglia can come together to form elaborate networks. And some of these elaborate networks can actually make our own decisions. One of the best examples of this is, well, and I guess we should give a name to this. These are what we call a plexus. One of the most elaborate plexes is our enteric plexus, or what is often sometimes referred to as our gut brain. That's not how you spell brain. Still not how you spell brain. There we go. You have that nice big cheeseburger for breakfast. Swallow it down into your stomach, and your stomach stretches in response to that cheeseburger. Is that the point where you consciously decide, all right, stomach, time to start working on that breakfast so I can get all the nutrients out of it? Is that what happens? No. What actually happens is, as the muscle stretches of the stomach, that sends a signal to this elaborate network of nerves in the stomach. And the stomach makes a very basic decision. I'm being stretched. That must mean there's food inside of me. So that means I need to start churning that food, and I need to release gastric juice to break that food down. And that whole decision-making process happens right there in the stomach. It has its own little gut brain where it makes that decision. And that's what these plexes are. So if we go back, we can see here. Again, we talked about the brain and spinal cord. We talked about our cranial nerves. We talked about our spinal nerves, our ganglia, and the sensory receptors, and even those multiple plexes. And of course, if we go back to our whiteboard, is there a relationship between our structural components and our functional components of the nervous system?
Yeah.
Yeah, absolutely. In fact, I'm going to cheat and move all of this down a little bit.
So for the structural components of the nervous system, are we just talking about the peripheral nervous system? Or are we
Nope.
also talking all for that and the central?
Well, well, I think you've hit on one of the keys that I'm trying to get to right now. Hold on.
Sorry.
No, I love that. Absolutely. So as you have aptly pointed out, these structural components are divided between the two main divisions of our nervous system. And as it turns out, those two main divisions of our nervous system, and we can cheat and put a nice little line like this here, are indeed what we call the central nervous system. The central nervous system is what is responsible for the integrative function. And the peripheral nervous system which, as we can see, is what carries information into and out of the central nervous system. And of the structures that we've talked about, which of these structures make up the central nervous system?
The brain and spinal cord.
Absolutely. Remember, the point of the central nervous system is to receive information, send information out, but also to make decisions. Your spinal cord does make decisions. As we talked about, when I put my hand on something hot, that decision doesn't go to my brain where I go, hey, I'm in pain, this hurts, I should do something about it. Yeah, that's a great idea. Maybe we should move our hand. Sure, that should be a great decision. Let's go ahead and do that. And then I send the signal to my hand and I pull it away. Is that what happens?
No.
No, as we talked about, that signal goes into my spinal cord, where my spinal cord makes the decision, hey, that's painful, pull away. And it goes, and I pull my hand away before the signals even reach my brain that I'm in pain. And as we talked about, what do we call those decisions that are made by the spinal cord?
Reflexes.
Yeah, they're reflexes, exactly. So our brain and our spinal cord are what make up the central nervous system, whereas our nerves, our ganglia, our special sensors, special sensory structures, so let's say it that way, and our plexus make up, let's make that a little smaller so it fits, make up our peripheral nervous system. All right. So far, so good?
Yeah.
Does seem to me I've left an awful lot of room here still on the board. And so let's talk about that for a second. When we discuss our sensory information that we're receiving, notice we said from inside and outside of the body, right? As we learned in skin, there are different types of sensory structures that can give us different types of sensory information. So not surprisingly, there are subdivisions. For our sensory branch of the nervous system. Those three subdivisions, I think I have enough room to do it a little bit bigger. The first, maybe we'll go A, B, and C so that we don't get confused with the numbers. The first is what we call somatic sensory. Somatic sensory is basically general information about the outside world. Actually, this isn't going to fit at this size. All right. What parts of your body can you perceive temperature from? Yeah, skin, but what parts of your skin? Just your right pinky finger? Not a trick question. Is your right pinky finger the only part of your skin that you can perceive temperature with? Yeah, no, Augustine's got it everywhere, absolutely. Right now, is everywhere equally sensitive to temperature? No, but everywhere can perceive temperature, right? Pressure, right? Tickle, itch, those types of are general sensory information that we can pretty much receive from all over our body. This is very different from our special sensory information. Special sensory information is specific information, typically from one specialized structure. How many different parts of your body can you perceive light from?
One.
One, just, well, technically two, hopefully, both eyeballs. But you have the right idea. And your eyeballs are only specialized to see light. I am not telling you to do this. In fact, I'm discouraging you from doing this. Do not do this. But up to this point, some of you may have made the errors of doing this before, because when you wake up in the morning, you need to wipe those eye boogers out of your eye. And if you rub your eye a little too aggressively, which again, I am telling you not to do because it can damage your retina. But if you were to push, theoretically, don't do it. But if you were to theoretically push a little hard on your eyeball, what happens?
You see light?
You see light, exactly. Is it because there's light present?
No.
No, it's because what you're doing is you're increasing the pressure inside your eyeball, the intraocular pressure, and that pushes on the retina, stimulating the retina. Now it can damage the retina, which is why you don't want to do it. But when you push that pressure onto the retina, it stimulates the retina, and the retina sends a signal to your brain. And because your retina is specialized for one type of information only, vision, you see a white light when that occurs. But notice still, this is information coming from outside of the body. So we also still need to have information from inside of our body, what we call our visceral or our autonomic sensory. And this is basically information about the internal environment of the body. Can you tell me right now what your internal body temperature is?
No.
No, if you could probably figure it out if you were able to go grab a thermometer or something like that. But what about your glucose levels?
No.
No, unless you're diabetic, then maybe you're able to tell me that. What about your calcium levels? Can anybody here tell me about a quart low of calcium right now? Just
No.
by the taste of the inside of your mouth? No, of course not. However, are there specialized structures inside of your body that is telling you exactly how much calcium is in your blood, or what the pH of the blood is, or what your internal temperature is, or how much sodium you have, or all of those types of things? Is that important information to have? Yeah, absolutely. So all of that is vitally, vitally important information to have. And we have these visceral or autonomic sensory that provide that internal information. They're actually special receptors that are known as interoreceptors. And these interoreceptors tell us what's going on the inside of our body. I don't like that big. Let's go ahead and go back there. All right. So those are our three subdivisions of our sensory. And if our sensory has subdivisions, do you think that our motor has subdivisions?
Yes.
Yeah. It has two subdivisions. And conveniently enough, these two subdivisions are divided by the effectors, which I've conveniently separated from each other. Notice, as we've already talked about, we have the branch that provides us the voluntary control of our skeletal muscle. And as you know from the previous section, what do we call that branch of the motor nervous system?
The somatic motor?
It was somatic motor, absolutely. Right, remember it was our somatic motor neurons that provided that information where we made the decision, our somatic motor neuron, right, formed our motor units. So that somatic motor neuron is part of the somatic motor branch, and it controls our skeletal muscle. You probably also know the name of the branch that controls our cardiac muscle, our smooth muscle, and our glands. This one provides involuntary control of our smooth, there you go, Mitch has got it, muscle, cardiac, muscle, and glands. Excellent. And we call this our autonomic motor. Excellent. And there are two subdivisions of our autonomic motor, which again, you might know as well. What are the two subdivisions of our autonomic motor? There we go. Excellent. Our sympathetic and our parasympathetic nervous systems. You may or may not have heard those terms before, but you probably know the fancy rhymes that go along with them. Our sympathetic is the one that helps us deal with that stressful situation, like that bear with an axe walking in the room. And what do we call that type of situation when a bear with an axe walks in the room?
Fight or flight.
Fight or flight. There you go. Perfect. And what's the cutesy mnemonic for our parasympathetic, which you'll all be using on Thanksgiving?
Rest and digest.
There you go. Exactly. There we go. As we will learn, fight and flight is actually a really, really good representation of what the sympathetic nervous system does. Rest and digest, not quite as good for the parasympathetic. We'll have some better ones that we'll use when we get there, but that's good enough for right now. So there you go. Those are the two subdivisions and the two subdivisions of the autonomic, breaking down all of the subdivisions of our motor branch of the nervous system. Again, we've done a really nice job of doing this with the colors, with the illustrations here on the whiteboard, but you'll notice that your book does a good job of this as well. Again, as we mentioned, we have our central nervous system, which is our integration, our command center, the brain and the spinal cord. Our peripheral nervous system, made up of the nerves, the ganglia, the sensory receptors, and let's not forget those plexes. Of course, a plexus is a combination of ganglia and nerves, so really it's not a new thing, it's just a name for a fancy bundle of all of these things. And again, our sensory is afferent, bringing information into the central nervous system, and motor is efferent, carrying information out of the central nervous system. As we also talked about, we have subdivisions, our autonomic or visceral sensory, our special receptors, and our somatic sensory, and subdivisions of our motor, our somatic motor, our autonomic motor, and then our fight and flight and rest and digest, our sympathetic and parasympathetic. And again, we've done this all here with words. Here is the great picture from your textbook, which does a good job of showing us almost everything. Notice the one thing this picture is missing is, for instance, a nice big eyeball to remind us that we have our special sensory coming in. So notice our sensory is blue coming in. It can either be somatic, like from the skin, special from the eye, or visceral or autonomic from the smooth muscle, cardiac muscle glands coming in. Here's our central nervous system made up of the brain and the spinal cord. whereas the peripheral nervous system is all those nerves and both cranial and spinal and ganglion and other such things. And notice with our motor out, we have our somatic motor that goes to our skeletal muscle, and we have our sympathetic and parasympathetic, which go to our smooth muscle, our cardiac muscle, or our glands. And again, for our autonomic, there are two branches, fight and flight and rest and digest. So notice here we're getting an increase in activity from our sympathetic to the heart in our fight or flight. Whereas with our rest and digest, which is involved in housekeeping, we get an increase into our bladder control. So here we see, with the exception of the eye, that nice visual representation that we just did on the whiteboard. All right. Questions on this? All right, excellent. We need to switch gears now and talk more about the tissues and the cells that are making up our nervous tissue. It's a little bit early, but this is a good natural stopping point. So I can leave you here with the picture, or I can leave you here with ours. Let's go ahead and leave it with ours. So I will go ahead and leave this on the board while we take our first break. Again, we'll take a 15-minute break, which means coming back at 9:02, and I will start the recording at that time. All right. Any questions on this structural and functional organization and all the subdivisions and components of this before we take our first break?
For the structural components of the nervous system, just the brain and the spinal cord
No.
belong to the central, or is that
No.
just for everything?
All of these things, the brain, the spinal cord, the nerves, the ganglia, the plexes, the special receptors, all of these are structures of the nervous system.
Okay.
Yeah.
Okay, thank you.
Yep. Right. The brain and the spinal cord are the only structures that make up the central nervous system. And then the nerves, the ganglia, and the plexes and the special receptors, remember, are what make up the peripheral nervous system. So the same way the functions are divided, the structures are divided as well.
Yeah, that was my question. Thank you.
Yep. All right, excellent. All right. I will see you guys at 9:02. All righty. So now that we've talked about the functional and structural organization and subdivisions of the nervous system, as I said, we need to talk a little bit about the nervous tissue. Nervous tissue, as I mentioned, is our fourth tissue type, and it is comprised of neurons and neuroglial cells. As we mentioned, the neurons are the functional cells. These are the ones that produce and spread action potentials. And as we saw back in the muscular system, these are the ones that then communicate to the target cell, telling them what to do. We saw how they, for instance, release that acetylcholine at the neuromuscular junction to stimulate the muscle cell to contract and to do its job. So those are our neurons. We've talked a little bit about that already. When we talk about their characteristics, they are similar to muscle cells in that they are irritable. And what did it mean to be irritable again?
To receive a stimulus and then respond to it.
Yeah. So when they are stimulated, oh, I like that reaction. That's a great way to describe it too. They are able to respond to a stimulus. When they're stimulated, they're able to respond. Like we saw with our muscle tissue, they respond by producing an electrical signal. So again, they're electrically excitable, just like we saw in our muscle cell. And like the muscle cell, they have conductivity. So not only can they produce that signal, but they can also spread the electrical signal long distances. I think we talked earlier in this class when we were talking about the muscular system, how if I had a, when we're in the classroom, I have a nice long wooden yardstick, and it's got the metal caps at the end. And so if I were to take that metal cap and jam it into a light socket, right, there would be smoke and we'd get a big electrical outburst and things like that. But me holding the other end of that would be perfectly fine, because that wood doesn't conduct the electrical signal. If I tried to take a metal yardstick and shove it in the light socket, then yeah, then what the rest of my hair that's left is going to stand on end as a result of it, because it has the ability to produce. So this neuron, again, some of these neurons, their axons, as we talked about, start in the small of your back and go all the way down to your big toe. They can be over 1 meter in length. And we're not even talking about basketball players where they're like, 70 feet tall. So these neurons can carry signals really, really long distances. In fact, if you had a neuron that went from here to Seattle, Washington, you could actually send an action potential from here all the way to Seattle, Washington. So it has that ability to spread those signals long distances. So very specialized cell, and we've talked a little bit about the anatomy, but we'll talk some more about it as well. And as we mentioned, the neuroglial are the support cells. However, notice there's an asterisk next to those support cells. How many people here have heard of an individual by the name of Albert Einstein?
Me.
Definitely.
Okay, a couple people, excellent, absolutely. For the rest of you, need to get out more. But Albert Einstein, for those of you know, incredibly smart individual who then died. And when he died, he donated his brain to science. And all of the neuroscientists were all a slather, because they were all excited about being able to cut up Albert Einstein's brain and see what kind of unique neural connections he had that allowed him to be able to make the massive leaps of knowledge and understanding that mere mortals like you and I cannot make. And so when they finally got his brain, and that's a whole big long story in and of itself, but when they finally got his brain, they chopped it up, they looked at the pieces, and what they found is that his neural network is exactly the same as yours and mine, which is a perfect opportunity to call up grandma, say, hey, grandma, guess what? I have the exact same neural network as Albert Einstein, and she'll be very impressed, and she'll send you 20 bucks in the mail. Now, that doesn't mean that his brain was identical to a typical brain. There were some regions of his brain that had as many as 4 times as many neuroglial cells as a typical brain does. This finding and other findings like this have shown that maybe these neuroglial cells do play more of an important role in processing and understanding and memory and things along those lines as well. And so over the past 10 years, there's been a dramatic increase in the study of these neuroglial cells to see if there's some big global over effect that they have on our processing of information. So should you take this class, I don't know, 5-10 years from now, and quite frankly, I don't know why you would, but if you would, we may be talking a completely different story about what these neuroglial cells do. But this is very, very new information. It's currently being researched. And so for right now, we'll focus on the things that we do know, the things that these neuroglial cells do to help to support our nervous function. But like I said, it is an interesting field that is currently being explored and may be expanded upon.
And so some people are just born with more neuroglial cells,
It's
or
not just a matter of more. Well, so that's a great question. We don't know whether he was born with more or whether he developed more. And it isn't just more in general, it's more in specific locations of the brain as well.
okay.
So, and again, you're looking at one static point in time. We don't know whether he started that way and that's why he turned out the way he was, or if it changed, modified based on something he did or what. I mean, there's so many questions to be answered, and we're just scratching the surface on that. But What we'll focus on is what we do know. One of the things we do know is what a typical neuron looks like. And as you see here, this looks pretty much like the drawings that I've been drawing for you. We have a nice big centrally located cell body. And of course, we can now give a name to this big centrally located cell body. It is a soma. Notice this soma has all of the things that we would expect to see. It has a nice big nucleus, and they've even put a nucleoli in here. They have some mitochondria in here. This clearly is representative of the Golgi apparatus and things along those lines. However, this is not entirely exactly like what a typical cell looks like. There is some stuff inside here known as Nissl bodies. When they first looked, and I'll use purple because that was the color that Nissl is. When they first started looking at these neurons and they used a special stain called the Nissl stain, what they found is that the cell was very, very darkly stained. There was a whole lot of something in here that showed up when they used the Nissl stain. And so without fully appreciating and understanding what it was, they just simply called it the Nissl bodies. However, as our microscopes got better and we got better at being able to explore and understand this information, what they found is that Nissl bodies are these large amounts of flattened membranous sacs with these tiny little bumps on their outer surface. What does that sound an awful lot like?
A rough endoplasmic reticulum.
Exactly. So this thing that they've been calling the Nissl bodies is actually, in fact, the rough endoplasmic reticulum. And this cell, the cell body of a typical neuron, has a massive amount. Why would it want to have a massive amount of rough endoplasmic reticulum? What does that rough endoplasmic reticulum do for us again?
Synthesizes proteins.
Exactly. And this isn't surprising because a neuron needs basically two main types of proteins. As we have talked about, it is going to communicate with its target cell, usually by making that neurotransmitter, like acetylcholine, that needs to be released. So that's a protein that we need to make a whole lot of to be able to release and communicate to the cells. But remember, our neuron, as this picture shows, and as we've talked about, has a lot of processes. And some of these neurons can run a meter long in their axon. So in that case, they're going to need structure. They're going to need some integrity. So we need to have a very stable cytoskeleton, right? That is going to provide that scaffolding, that structure, that shape to this. And in the axon, it will also provide our pathway, our highway for transmission of that neurotransmitter. We can use those motors to send the neurotransmitter from one side of the cell to another. And so these big, elaborate cytoskeletal structures, of course, also have a fancy name. They are protein filaments inside of a neuron, so they're called neurofilaments. And so again, if you look at our illustration here, I'll change the color to green. You can see these linear components that they're drawing here are supposed to represent that neurofilament, that cytoskeletal structure that is going to help to give the shape to the axon and the shape to the dendrites and shape to the neuron itself. So again, we have a typical cell body like we expect with some specializations to it. But we also have two types of processes. Again, as we learned from bones, processes are things that stick out. A typical neuron has multiple dendrites. These, as we've mentioned, and then we see from the picture, are short and they're elaborately branched. And their job is to receive information. A single neuron can receive information from as many as 10,000 other neurons. So 10,000 other neurons could be sending information to this neuron to help it decide what it wants to do. So having all of these elaborate branches allows it to receive that information. And all neurons have one long, non-branching axon. This axon has two things. It carries information away from the cell body. And it's the axon that is specialized to produce and spread the action potential. Now, if you remember, when we talked about our muscle cell, and our muscle cell's plasma membrane was specialized for producing and spreading an action potential, so we gave it a special name. And what was the special name we gave to the plasma membrane of our muscle cell?
A sarcoplasm? Or
Sarcoplasm was on the inside, but the plasma membrane was the sarcolemma. Excellent. So sarcolemma is what we call the plasma membrane of our muscle cell, specialized to make and spread an action potential. So the specialized plasma membrane of the axon, axolemma. So the specialized plasma membrane of the axon, plasma membrane of the axon, that allows it to produce its action potential, we call an axolemma.
Is this picture in our book? Because we don't have it on our slides. We have a different picture. So I was just wondering.
Yeah, this picture is
Okay.
from the textbook.
Okay, thank you.
Now, I need to erase some of my drawing here. So let's get rid of this. So notice we have a bit of an issue here. We know our axon's plasma membrane is specialized to produce and spread an action potential. And the fact that I'm saying that means that the plasma membrane of our cell body and the dendrites is not specialized to produce an action potential. So do you think that there's just a transition point where instantly it goes from being specialized to being non-specialized?
No.
No, as it turns out, there is this, and I guess since it's in between red and blue, I guess I have to make it purple. There is a transition region where the cell plasma membrane gets a little more specialized, and then a little more, and then a little more, and then a little more, and a little more, until it's fully specialized. And this transition region where the axon connects to the cell body is known as the axon hillock. Now, do you think making one tiny change to the axon, to the plasma membrane, is going to make it be able to produce an action potential? One tiny change. Do you think that's all it takes? No, of course not. However, do you think it needs to be 100% changed to be able to produce an action potential? What if it's 99%? Do you think if it's 99%, it can produce an action potential?
Yeah, I could.
Yeah,
Yeah.
95 probably, right? But there is going to be some point where suddenly it goes from not being able to produce an action potential to suddenly it's changed enough to produce an action potential. And that specific spot here in the axon hillock is what we call the trigger zone. Now, do you think with a naked eye, or even for that matter with an electron microscope, we can look at the axon hillock and see exactly where that trigger zone is going to be?
No.
No, the trigger zone is not something we can see. We just know two things about it. We know it is inside the axon hillock, and we know that that is going to be the first place where we can produce an action potential. So as we look at that axon hillock, inside that axon hillock is a trigger zone. And that trigger zone, as I mentioned, is going to be the first place and produce an action potential. Excellent. All right. Questions on that? All right, that's all the new stuff, because if we look at the other end of our neuron, oops, wrong button. We've seen the other end of our neuron before. We talked about this with our muscle cell. We know that we have a single unbranching axon that comes down, and as it nears the target cells, as we mentioned before, it can branch. Notice here, your textbook uses the fancy term teledendra to identify those, but remember, we called them axon terminals. Either name is acceptable. In fact, there's probably 4 or 5 different names that have been given to this particular structure, but to stay consistent with what we used in the last section, we can use the term axon terminals. And as you know, notice here, the end of our axon is specialized. Notice your textbook uses the term synaptic terminals, but what did we call the specialized end of the axon?
Synaptic end bulbs.
Exactly.
So are they interchangeable? Or
Yes.
do we use, okay.
And like I said, there are, for this one too, there's probably easily a half dozen different terms. Synaptic terminals, synaptic boutons, terminal buttons, synaptic end bulb. All of them are anatomically appropriate. But again, for consistency and so that we don't have any confusion, the terms I will continue to use are axon terminals and synaptic end bulb. But you, like any time we've talked in this class, you may use any appropriate anatomical term when describing these things. And since we just finished doing it, remind me that our synaptic end bulb basically has 2 main specializations. What are the 2 main specializations of my synaptic end bulb?
The voltage gated calcium channels.
Excellent. Voltage gated calcium channels, excellent, which allow calcium to come in, allowing our cell to do a wonky thing. And what is the wonky thing that this cell will do here at the synaptic end bulb? What's the other specialization? What else did we find down in the synaptic end bulb?
The neurotransmitter acetylcholine.
There we go. Vesicles containing neurotransmitter. Yes, you are correct. The one we've talked about at the neuromuscular junction was acetylcholine, but there are over 100 neurotransmitters that have been identified in the body. So remember, neurotransmitter was the generic term we used. And since we're talking about a typical neuron, we'll just use the term neurotransmitter. So here in the synaptic end bulb, we have vesicles containing neurotransmitter to allow us to communicate with whatever our target cell is. And then obviously we don't want to be releasing that neurotransmitter all the time. So how do we control the release of it? With our voltage gated calcium channels. Excellent. Questions on that? All right, I have a question then. Notice I said typical neuron, not all neurons, typical neurons. So what does that imply?
There are other types.
There are other types, absolutely. And not surprisingly, when we talk about I want to do this one. Let's do it this way. How about fancy? When we talk about neurons, there are actually two ways we can distinguish our neurons. What do you think the two ways we can distinguish our neurons are?
Structurally and functionally.
There you go. We can have our functional divisions of neurons. And as it turns out, there are 3 functional divisions of our neurons. And we can have structural divisions of our neurons. And conveniently enough, there are 3 of those as well. Of course, when we talk about our functional divisions, this is going to basically be related to the direction they carry information. And structurally, not surprisingly, is going to be essentially the shape. Or we can actually be more specific in this case with our shape. It's really about the number of processes. And lastly, do you think that there is a relationship between function and structure?
Yes.
Yes, of course there is. Excellent. Let's talk about our functional divisions. And the good news is these functional divisions are pretty much similar to what we saw in our functional divisions of the nervous system. We have sensory neurons. And guess which direction sensory neurons carry information? Well, away from what? Or into what? So away from
Into
something
the-
or into something?
I can't say into the integration system or
Close, Augustina. Again,
Okay.
sensory, that when I see that bear with an axe, I want that information to go into the brain, right? So I want to be able to tell the brain about it. So sensory information, sensory neurons carry information into the central nervous system. So then, not surprisingly, our third functional division is going to be motor neurons. And which way do motor neurons carry information?
out of the central nervous system.
And if you also remember, we had fancy names for this directional terms. Carrying information into the central nervous system, we said it was what kind of a pathway?
Afferent.
Afferent. And again, afferent obviously is the right way to say it, but I emphasize the A because it helps me to remember how to spell it. Afferent pathway. Whereas coming out of the central nervous system then was what?
Efferent.
Excellent. Excellent. So we have into the central nervous system, out of the central nervous system. So what's left?
Within the central
Yeah,
nervous system.
Absolutely. And the ones that carry information within the central nervous system, we call interneurons. So as their name indicates, interneurons carry information within the central nervous system. We could also think of them as being in between the sensory and the motor, right? Because they are inside the central nervous system. There you go. Hopefully that's not too tricky. Makes sense. And really not really new information. I guess the term interneuron is new, but we've kind of already talked about the three ways you could carry information into the central nervous system, out of the central nervous system, or the processing that takes place within the central nervous system. So that brings us to our structural divisions of the neurons. And notice again, as we mentioned, this is going to be named based on the number of processes. So one type of neuron, and let's go with brown for the cell body. One neuron has a centrally located, actually, let me do this. A centrally located cell body. It has one dendrite where it receives information. And this dendrite can be, oh, I'm doing circles, that's why it looks so weird. Bonds. Can receive information. And then it has one axon that carries the information out. And as we know, that axon can branch to form axon terminals, which can branch as well. So notice in this case, we have one dendrite. We have one axon. And a centrally located cell body. Now, as we have already talked about, our neuron's job is to receive information from the dendrite and carry that information out through the cell body to the axon. So notice information is traveling. No, don't want that to be purple. I mean, don't want that to be blue. I want it to be purple. Notice in this case, our information travels down the screen. And since we identify our neurons based on the number of processes, this one happens to have two processes. So since it has two processes, guess how we identify this particular neuron?
By.
Exactly. It is a bipolar neuron. All right. That makes sense?
Mm-hmm.
Excellent. Let's look at a second type of neuron. In this case, the cell body is actually located to the side. So it is a peripheral cell body. Connected to which is the axon. The axon is connected off to the side. So we have one axon. We always have one axon. Now, notice this is still going to need to receive information. But in this case, the dendrites will actually connect directly to the axon.
So is it considered one dendrite or no?
Well, again, it can be a dendritic tree. It can have multiple, the same way our axon has terminals, our dendrites can receive multiple information that it's bringing in. So again, this is considered, you are right, it is considered one dendrite, but So our dendrite connects to the axon. But more importantly, the dendrite doesn't connect to the cell body,
Oh, okay.
right? So since we identify these neurons based on the number of processes coming off of the cell body, how would we identify this neuron?
Unipolar.
Yeah, this one's going to be unipolar. Excellent. Notice neither of these looked like what we called a typical neuron. So let's draw that typical neuron. A centrally located cell body. Two or more dendrites. That attach directly to our cell body. And again, as we talked about, they're often branching. And how many axons?
One.
One axon, which of course we know can branch to form axon terminals, but we still have one axon. So notice this one doesn't have one process coming off the cell body, doesn't have two processes coming off the cell body, but it has many processes coming off the cell body. So what do we call a neuron that has many processes coming off the cell body?
Multipolar.
Exactly. And since we said this is what a typical neuron looked like, which of these three do you think are the most common? Multipolar, unipolar, or bipolar? Multipolar, absolutely. This is the most common structural type. All right. Since we're talking about most common, do you think we have an equal number of sensory, interneuron, and motor neurons in our nervous system?
No.
Where do you think there are more neurons? Are there more neurons carrying information into our central nervous system, carrying information out of our central nervous system, or within our central nervous system?
With within and without both.
Well, but you only can pick one. So is it
within?
going into the brain and spinal cord, coming out of the brain and spinal cord, or do you think the brain and spinal cord have the most neurons in it?
Most neurons inside of it.
Yeah, absolutely. In fact, over 90% of your neurons are in your brain and spinal cord. So our most common functional type are the interneurons. Excellent. All right. Questions on our functional or our structural divisions? The functional types of neurons or the structural types of neurons? Any questions on these three divisions, or six, I guess, divisions? Because we need to do one more thing, relate them to each other. Now, let's think about this. If interneurons are the most common type, if I were to randomly reach into your brain or take all of the neurons out of your body, put it in a bucket and pull it out. If the most common functional type is an interneuron, what do you think the structural classification
Multipolar.
Of our interneurons are going to be? Exactly. All of our interneurons, or most of our interneurons, all of our interneurons are going to be multipolar.
So does that mean the most common functional for the multipolar would be interneurons?
Yeah. Excellent. For our sensory, most of our sensory neurons are unipolar. So of course that makes it easy. Structurally, our motor neurons are what? Exactly. Mitch has got it on the head. Multipolar. Right. Remember when we were talking about our muscle cell, we drew that neuron with its multiple dendrites and its single axon coming and communicating with our skeletal muscle fiber. So clearly it's multipolar, right? And that what Mitch said, oh wait, you said bipolar. Uh-oh. Did I mean that I messed up?
No.
No, definitely not above messing up. I do mess up, but it turns out that functionally, indeed, both our interneurons and our motor neurons are multipolar. So then what the heck did the bipolar do?
Are they with the sensory neurons because you said most?
There you go.
Are
See,
unipolar?
catching up on all of my tricks. I love that. Absolutely. Notice we use that keyword most. And because as it turns out, from a functional standpoint, bipolar neurons are only found in our special senses. They're only found associated with our eye, for instance, or our hearing, or our equilibrium and balance, smell, taste. All those special senses we've talked about, all of them have bipolar neurons. So structurally, most of our sensory neurons are unipolar, but our special senses use bipolar. And again, I've done a truly amazing job of drawing this, but let's look at the pretty pictures from your textbook. Here we see an illustration of a bipolar neuron with its centrally located cell body, one dendrite and one axon. And notice it's found in those areas associated with our special sensory information. Here's that unipolar, again, a laterally, peripherally located cell body, one single axon, and then the dendrites up here at the top. And those are most of our sensory. And there is that typical multipolar that we saw, that typical neuron, multiple dendrites, one axon. This is the one we've been drawing up to this point of time. And functionally, these are our motor neurons and our interneurons. Now, there are two more points that I want to make. You are not required to use this term, but sometimes in the literature, instead of unipolar, you will see the term, oops, pseudo unipolar. And as we know, what does pseudo mean?
False or fake?
Yeah, false or fake, exactly. The reason for this is from a developmental standpoint, what sometimes happens with some of these unipolar neurons is that when the processes first grow out, one process grows out and grows this way. A second axon grows out and grows this way. And then as the cell starts to mature, these two bundle together into a single structure. So technically it was first made by two processes coming out. So it's kind of a fake unipolar. But since it functions the exact same way, and for all intents and purposes, when it's a mature adult cell, it's just the one process coming out, I have no problem with you just using the term unipolar. I just don't want you to be confused when you see the term pseudo unipolar. All right, so you may see that term, feel free to ignore it. It just means that developmentally, it started as two processes that fused together into one. All right. That's the first thing. Second thing, there are these three main structural classifications. However, do you think every single multipolar neuron in your body looks exactly like this one?
No.
No, of course not. There are dozens of different types. We did that. Here we go. Here, for example, are two of the dozens of examples of multipolar neurons. Notice both of them have a centrally located cell body, one single axon that comes off, and multiple dendrites that come off of them. However, notice they're very, very different. Notice this one over here has very long, very thin dendrites and a triangle or almost pyramid-shaped body, which is why it gets the name pyramidal cells. These happen to be the types of neurons you find in your cerebral cortex, right? A part of the brain that we are going to talk about a little bit later. Whereas this one over here, notice its dendrites almost form this kind of funny fan-looking appearance. And again, I'm sure it's just our illustrator that makes it look so two-dimensional in this shape. Well, it turns out, no, this is actually what this cell looks like. If you were to take this cell and rotate it 90 degrees, you would see that all of the dendrites really do fan out all on one plane. So it's not like a tree where they're going in all different directions. All of these dendrites line up in a single straight line. These particular ones happen to be found in the cerebellum, another part of the brain that we will talk about in just a minute. And these happen to be called Purkinje cells. Guess why they're called Purkinje cells?
Because he founded it.
There you go. Good old Bob Purkinje is an incredibly famous neuroanatomist. He loved the microscope, and he was actually a really amazing artist. So what he would do is he would get tissues, not just of the brain, but the entire body, and look at these under microscopes and find things that were really interesting. And he would then draw them. And of course, then publish them in a book and stamp his name on them. So
He
here
has his name on a lot of stuff, right? Like even like the last unit we had, I think Purkinje fibers or something.
Well, Purkinje fibers you're going to find in the heart. So we'll talk about
Right.
Purkinje fibers in the heart. Purkinje cells are going to be located here in the cerebellum. Yeah, Purkinje was an anatomy whore. There are 4 or 5 different things in the body that have the name Purkinje on them because he was the first to describe it and put his name on it. Absolutely. So yeah, absolutely. So again, we'll see numerous instances of Purkinje as we work our way through the body. All right. Which brings us to the last big concept that I want to talk about in our introduction here. One of the things we have been harping on since the very beginning of this class is that there is a relationship between structure and function. We've been harping on that absolutely since the very beginning of this class. However, one of the things that is going to be vitally important to understand about the nervous system is in the nervous system, there is a relationship between structure and function and location. All right. Here we see 2 perfect examples. 2 different locations, the cerebellum and the cerebral cortex, and they have 2 different types of cells, right? Does this part of my brain right here do the exact same thing as this part of my brain up here?
No.
No. This may move my face. This may move my hand, right? And if this moves my hand over here, does this move the same hand?
No.
No. So not only can there be differences in the locations within the side, but there can be lateralization as well, where what something does on one side isn't the same as what it does on the other. For instance, over here, I may move my left hand. Over here, I may move my right hand. But if I move a little bit more forward, right here is the part that allows me to speak. Whereas right here is the part that allows me to do math. So notice, just because they're different parts of the brain do different things, but also what's on the right side can be different from what's on the left side. So one of these things we're going to be strongly emphasizing in this section is the relationship between structure, function, and location. And the good thing about that is if you can figure one of those 3 things out, then you know the other 2. Right? If I show you a cell like this Purkinje cell, and you know it is in the cerebellum, even if you can't tell anything else about it, you know that that cerebellum is in the brain of the central nervous system. So functionally, what must it be? Well, if it's in the central nervous system, can it be bringing information into the central nervous system?
Is it integrative?
It's an interneuron. Exactly. Its structural class, a functional classification is an interneuron. And what must, if it's an interneuron and it's in the central nervous system, what must its structural classification be?
Multipolar?
Yeah. So Purkinje cell in the cerebellum, interneuron, multipolar. Those 3 things go together. Let's do that. Actually, questions on this before I add? Questions on our structural classifications or our functional classifications? All right, let's actually see what some of this stuff looks like. Here is the brain and a little bit of the spinal cord in this simple illustration. For those of you who do not have as much familiarity with this, as we were just talking about, remember those pyramidal cells are found in what we call the cerebral cortex. That is basically this big, huge mitten-shaped structure. It always reminds me of a big mitten because we have the big hand part there and then the little part for the thumb. So that is this region here is our cerebrum. And as we mentioned, this is where we're going to find those pyramidal cells. But here in the very back and posterior and inferior portion, this little stripe looking structure that we'll look at in just a second here, this down here is, as I mentioned, the cerebellum. So let's start first at the cerebellum. Here we see our cerebellum. Now, there's one more piece of information. So let's go back up sheet and I'm gonna go ahead and put this over here for right now. Oops, no, I want this to be white. The cerebellum is part of the brain. And so as part of the brain, what is the structural classifications of the neurons?
Of all neurons or just this one?
In the central nervous system. What is the structural classification of the neurons we will find in the central nervous system?
Multiple?
Yeah. They're going to be multipolar neurons. And what are they going to be functionally?
The interneurons and motor neurons?
Now, if they're in the central nervous system, they are interneurons. Okay. And they're multipolar. So notice, as we've seen before, they're going to be made up of cell bodies and dendrites and axons. Now, one of the things we haven't had a chance to talk about yet, but we are going to very, very quickly, is that axons have special glial cells that wrap around them, and they wrap their cell bodies hundreds of times around them. And what is the plasma membrane made up of? Come on. Think back to a cell. What is the plasma membrane of a cell? Phospholipids, fats. These fats wrap hundreds of times around the axon, forming a special structure called a myelin sheath. But the myelin sheath is basically just a fancy term for many layers of fat. And when you look at that fat on that steak you bring home to be able to cook, or you bring that pot roast out that you're getting ready to put in and you want to cut the fat off of it, what does the fat look like when you pull that meat out of the fridge?
Isn't it white?
Yeah, it's white. Absolutely. So the same way, if you remember in our peripheral, actually, let's do it. Well, I'll do it this way. If you remember in our peripheral nervous system, we had fancy names for a cluster of cell bodies and dendrites. We called them a ganglion. And an axon we called a nerve. So in our peripheral nervous system, we had names for clusters of cell bodies and dendrites and clusters of axons. Well, it turns out we have the same thing in our central nervous system. In our central nervous system, bundles of bundles of axons are called tracts, or they're called white matter because of the white color, thanks to that myelin, that fat. Our clusters of cell bodies and dendrites are known as nuclei, or nucleus singular, nuclei plural. Plural. Or since we did nucleus, since we did.
Is it also gray matter too? Or no, just.
Exactly, or gray matter. And that's what we see here. In our cerebellum, we have white matter, which is deep, and we have gray matter, which is superficial. And here in the cerebellum, we have something special that we don't see anywhere else in the nervous system. We have not one, but two layers of gray matter. So if you look here, I'll do it this way. One layer of gray matter, two layers of gray matter. These happen to be called the molecular and the cellular layers. You don't have to worry about that distinction. But we have those two layers of gray matter, and then deep, we have the one layer of white matter. And so you notice that continues on to the other side. So again, this gray matter wraps around the white matter in this case. So when you see these three layers with these nooks and crannies, this is a dead giveaway you were looking at the cerebellum. Now, again, we know our anatomists love labels. So let's work on some of these labels. And actually, I'm going to go ahead and keep stealing space from this. Notice that on our cerebellum, we have some raised ridges. And then we have those grooves in between. Now, are we going to get away with terms like raised ridges and grooves in between?
No.
No, of course not. So our name for these raised ridges that we see are folia. The singular, folium, or the plural, folia. They do this because it kind of looks like, when you look at it at a low magnification, it kind of looks like the edges of a leaf. So the raised ridges are folia or folium. And then the grooves is a term we've already heard before, sulcus. Just like if you remember, the intertubercular sulcus was the name we gave for that groove. Here, again, sulcus is the singular, sulci would be the plural. All right. Have I lost you yet?
No.
Excellent. Okay, perfect. Next, I want you to go back to the orange. Notice this deep white matter from all these folia come together, forming a very elaborate tree-like structure. And so when you cut through the cerebellum, this has a very distinct, very fancy shape to it. And so collectively, all the white matter of the cerebellum has a fancy name. It looks like an elaborate tree, and this is an elaborate tree that obviously would help to keep you alive. So this fancy name is basically the tree of life. Of course, we're not going to get away with calling it the tree of life. They have to make it fancy. So of course they used Latin. So what is the fancy name for this elaborate network of white matter in our cerebellum? Give you a hint. If you haven't printed out your study guide for the anatomy list for your brain and spinal cord, this is probably a good time to look at that. Tree of life in Latin. No one's taken Latin?
No.
Arbor vitae. It is known as the arbor vitae. So notice these are all things that I could ask you on the exam. On the exam, identify the specific matter, white matter. There you go. Identify the name for this structure, arbor vitae, right? I could take my arrow, identify this groove / space. What would you say?
Puss.
Sulcus. Identify this ridge.
Folio.
Folium.
Excellent, right? Identify the region of the body or the specimen that we're looking at here.
Cerebellum.
Yeah, cerebellum. Excellent. So those are all good things. But remember, the whole reason we came here to begin with was because of those Purkinje cells. We came because we wanted to find those Purkinje cells. And it turns out that even at this low magnification, because it's slightly out of focus, can't see them, but I can tell you exactly where they're going to be. All of the cell bodies of your Purkinje fibers are all going to be found right here on the border between the two layers of gray matter. So in these big clusters in these regions, right in between the 1-2 layers of gray matter, that is exactly where you're going to find these Purkinje cells.
So can you ask us where, like, what would be found here?
So notice on the exam, I could point right here and ask you to identify the specific cell that would be found in this location. And what would you say?
Purkinje cells.
Excellent. What is its structural classification? Multipolar. What is its functional classification?
Interneurons.
Interneuron. Notice all three things went together. You know the location. You recognize this as the cerebellum. And because you knew the location, you knew the structural classification, you knew the functional classification. Now, technically I could do that here, but I don't have to be that challenging. Notice if we increase the magnification, notice different stain. Remember, different stains change the color, and the color is not as important, but we still see a folia. We still see a sulcus. We see another folia. Notice I'll use, no, not light. I'll use, see if the dark red shows up. Again, we see that little bit of gray matter that is deep. And then we also see not just 1, but 2 layers of gray matter. Not just 1, but 2 layers of gray matter. And notice, lo and behold, right at that border, we see all of these cell bodies. So notice I don't even have to say, what cell would you find here? I could have this picture, point this nice big arrow right here, and ask those same 3 questions. Identify the cell, the specific cell.
Purkinje cells.
Identify the structural classification of this cell.
Multipolar.
Identify the functional classification of this cell.
Interneurons.
Yeah. Notice you really can't tell that by looking at this. Looking at this cell, you can't see the processes because they didn't stain well. But remember, as we talked about, this is going to have a big elaborate dendritic tree that's going to come out here like this, and then a single axon that's going to come in and become part of the white matter. So notice, even though we don't see the processes, just because we know our location, we know our structure and our function. However, I don't want you to think I'm lying to you. So, oh, here's some more great up close. Now notice this one, we don't see the white matter. So I could not give you this one on the test because you can't really tell at this high of a magnification where we are in the body. But notice you can start to see some of the processes that are coming off, some of the dendrites that are coming off. We don't see it on all of them, but some of them we can. But what's really cool with some of these electron microscopes, there it is. With these electron microscopes, they're actually able to put a fluorescent dye into a single cell so we can see all the parts of it. And so lo and behold, here we see, okay, so many colors, I don't know what I can use. I wonder if white stands out. Let's try white. Here we see a cell body right between the two layers of gray matter. Here we see the white matter, the axons there. And notice again, here we have these elaborate fan-like dendritic trees that are going off big into the gray matter. And we don't really see the axon coming off of it. But again, remember, this is a random cut through this cell. We throw this cell up, if we throw an apple up in the air and with our eyes closed, we slice it. What are the chances that when I slice that, I'm going to perfectly see the stem?
Oh, really small.
Really, really small. So is it terribly surprising that we don't actually see the axon coming off of any of these?
No.
No, not really, because it's not likely that we're going to see that one process coming off. But we know it would come off and go and become part of that. So again, this is a beautiful example of these Purkinje cells. And again, location, structure, function, all go hand in hand and hand.
So the middle is the white matter, and then the bluish and green is the gray matter. And this,
yes, correct.
Okay.
Yeah, for this particular coloration. And again, notice we're basically looking at one folia. Right?
Oh,
We don't see the top of the folia, but we're seeing there is a cap obviously to it at some point. Again, it's a very, very high magnification view. I wouldn't necessarily give you this by itself naively, but now that we've spent some time looking at it, we can make sense of it. But basically, let's go back. What we are looking at right here in a very up close view. Oops, wrong buttons. If you notice, is basically that. Right? Or let's even orient it closer to the same way. Basically, not quite the right orientation, but basically we were looking at something like this. We had the white matter in the middle, the two layers of gray matter on the side, and then the Purkinje cells with their branches coming off this way.
and so how would we know when you want us to state it's white matter versus the arbor vitae? Because
well, if I'm asking for matter, then give me white matter. If I'm asking for the specific name, and again, remember, this is the only place where it's arbor vitae. So this is the only thing that has a special name for its white matter, really.
Okay.
Okay. Excellent. All right. So a location, a structure, and a function all go hand in hand, and a specific name for a cell that we found there. Here is our spinal cord. Again, we'll get to the anatomy of some of these things in more detail, and the spinal cord is a perfect example of this. What I want you to see, though, right off the bat, and the reason we're looking at it is twofold. For starters, it's this kind of oval shaped structure with this pretty little butterfly in the center of it.
So is this like a cross section?
Yep, this is a cross section through the spinal cord. Now, spinal cord, central nervous system or peripheral nervous system?
Central.
Yeah, this is indeed part of the central nervous system. Excellent. And what's interesting about this is notice that it has its gray matter is deep. And in this case, the white matter is superficial. This is also part of the central nervous system, but notice this is the opposite of what we saw in the cerebellum. In the cerebellum, the gray matter was on the outside, the white matter is on the inside. So again, remember what that means is this is where the axons are located, and this is where the cell bodies and the dendrites are located. All right, you with me so far?
Yeah.
Excellent. This happens to be the front of the spinal cord, and this happens to be the back of the spinal cord. And I make a point of emphasizing that because notice that our butterfly here in the center, the gray matter isn't evenly distributed in all the regions. There is much more gray matter here in the front. And this big, large piece of gray matter is actually called, because it's in the front, the anterior gray horn. All right. Now, notice here we're at a very low magnification. But even at this very low magnification, we see tons of dots located here in the anterior gray horn, a location. Turns out these neurons have axons that travel out the spinal cord and go and communicate with our skeletal muscle cells. So since these neurons communicate with skeletal muscle cells, what is their functional classification?
Motor.
These are, and we could, okay, you get partial credit for motor. Could we be slightly more specific?
Motor neurons.
What type of motor? There we go, Mitch. Somatic motor. And if they're somatic motor, what must their structural classification be?
Multipolar.
Exactly. So in here, they're going to have a centrally located cell body and little dendrites, multiple dendrites that come off of it. Notice once again, we have a location, anterior gray horn. We have a function, somatic motor, and we have a structure, multipolar. So structure, function, location go hand in hand in hand. Now, this is a beautiful cross section of our spinal cord, but it's not the only way we can look at the spinal cord under the microscope. With the spinal cord, we can do a particular prep called a spinal cord smear. And guess how you make a spinal cord smear?
Smearing the spinal cord.
Yeah, you take your slide, you take your spinal cord, and you smear it on there. Now, if you do that, is it going to have this beautiful butterfly shape to it?
No.
No, it's going to break up the tissue. But the nice thing about breaking up the tissue is that allows us to see the cells more clearly. So we have a location. This is the spinal cord. And more specifically, most of the neurons in the spinal cord, remember, are in the anterior gray horn. So when we look at these neurons, they're most likely from the anterior gray horn. And as we look at, for instance, this one right here, we see our centrally located cell body with its nucleus and actually some nucleoli inside of there. And I count 1, 2, 3, 4, 5, at least 6 processes that are coming off of it. Heck, even this one with 3 coming off, 1, 2, 3, although it looks like that might have been broken. Maybe there's another one there. Maybe there's another one there. But even with just those 3, as I look at this, is it easy to tell whether these are, which one's the axon and which ones are the dendrites?
No,
No. Do I expect you to be able to do it?
no,
No. But can you count?
yes,
Yes. And once I get to 3 processes, do I need to keep counting?
no,
No. Once I get to 3 or more processes, what must the structural classification be?
multipolar,
Excellent. And this multipolar neuron from the anterior gray horn, what is its functional classification?
somatic motor.
Somatic motor.
In the slides or in our whiteboard notes, you said 2 or more dendrites. Is it 3 or more then?
No, because you have 2 or more dendrites and an axon.
Right.
So notice if you get to 3 processes, even
Oh,
if you
with the
can't tell
axon,
what
okay.
they are, you
Okay.
know that it must be multipolar.
Okay, thank you.
Yep. Now, one more thing. Any idea what all these little dots might be? Is that just dirt that got on the slide? An intern did this very messily. Close. Ganglia is not a good guess. There you go. These are the support cells. These are those glial, or you may also use the term neuroglial. Those terms are interchangeable. Glial or neuroglial cells. Over 100 different types of neuroglial cells have been identified in the nervous tissue. Have I given you the names of any of them yet?
No.
No. You are going to have to learn, I think it's like 6 or 7, something like that. But even if you knew the names of those 6 or 7, by just looking at those dots, can you tell what they are?
No.
No. So again, if I point at one of these tiny dots, you just have to use the generic and say that it is one of those support cells. So that would be a neuroglial cell or glial. Again, both would be acceptable ways of identifying it. And we don't have to tell which one it is. In fact, there's no way to know. I don't know which one that is. So if I don't know, you certainly don't know. And so we're not going to worry about it. So the same way we don't have to worry about any of these things. If I point at this, that's just a process, right? And these are just neuroglial cells.
So I know this may be a little early to ask this, but splitting up the lab exam, is there just going to be a lot of histology and I guess anatomy of?
There's not quite as much histology as in the last section. But no, here's what I would say. 50% of your lab exam is going to be your central nervous system anatomy handout and the histology that goes along with that. So the stuff we're doing right now, that'll easily be 50% of your lab exam. The remaining 50% is going to be divided between our cranial nerves, our autonomic nervous system, and our sensory information. And like I said, depending on if we run late, like we seem to always do, if we run late during the section, so there's so much to cover, you won't really have any sensory that you're going to be responsible for on a lab exam. So that would mean basically 25% autonomic nervous system and 25% cranial nerves. So that's roughly how it'll break down.
Thank you.
And any sensory we get in means less cranial nerves and less autonomic. But I don't know if the past 2 semesters we've been online, I haven't gotten to any of the sensory stuff. Maybe we will, but I don't know if we'll get enough where we have to worry about the anatomy of it for it. So we'll see how we get. All right.
Thank you.
Perfect. So notice another location structure function relationship. Oh, excellent. Notice here, even at a higher magnification, can we tell what these individual neuroglial cells are?
No.
No. So these are just going to be neuroglial cells. And again, might it be possible to tell which processes are the axons and which processes are the dendrites? Maybe, but do I
No.
expect you to be able to do it? I mean, some are easy. This really small one, do you think this really small one is likely to be the axon?
No.
No.
Probably not. This one looks the biggest, so maybe that's the axon, but this one looks pretty big as well. So again, I'm not going to hold you responsible. We just need to know processes. But notice there are a couple other things that we can see here. Notice we can very, very clearly see the nucleus here and the dark stained nucleoli inside of there. But remember, as we talked about before, there's all this shaded material. What is this shaded substance again? This dark stained substance that we're seeing inside of here?
The missile bodies.
The Nissl bodies, excellent. Is there anything else we could correctly call it on the exam?
The rough endoplasmic reticulum
Yeah.
with free floating ribosomes.
Exactly. Well, yeah, you don't even have to put free floating ribosomes, but absolutely.
Okay.
Yeah. This is the rough endoplasmic reticulum or the Nissl bodies. Either would be a fine way of being able to describe that. Remember, the vesicles aren't going to be until we get to the axon terminals at the end of it. But notice there's one other thing that can be seen in here as well. Notice when we look in the processes, there are these linear structures, these stabilizing structures, this scaffolding that we can see in the processes. And what would those linear structures be?
Neurofilaments.
The neurofilaments. Exactly. So notice when we look up close, we can actually see the Nissl body or rough endoplasmic reticulum and the neurofilaments. All right, excellent. All right, back to our spinal cord intact. And notice out here to the side, so notice this is out here in the peripheral nervous system. Is a big bundle. And actually, let me draw my circle more clearly. This is a bundle. Notice, and I'll use a different color for this. We have the axons coming out and the axons coming out of the spinal cord. These come together to form our spinal nerve that is going to travel off, and we actually see the spinal nerve on the other, well, actually, no, the spinal nerve would come off on the other side there as well. So that's where the spinal nerve comes off from the axons. But this that here that I've colored in green, this enlarged mass is a cluster of cell bodies. Now, this cluster of cell bodies happens to be in the peripheral nervous system. So what was the fancy term we gave for a cluster of cell bodies in the peripheral nervous system?
Is it plexus? Plexus?
No, plexus is a combination of nerves and these bundles.
Was it ganglia?
Ganglion, absolutely. This is a ganglion. Now, notice this ganglion is connected to the axons that come out of the back of the spinal cord. This structure that comes out of the back of the spinal cord, these axons, are called the dorsal root. So guess what we call the cluster of cell bodies that sits on the dorsal root? Not a trick question.
Ganglion dorsal root.
It's the dorsal root ganglion. Now, notice I keep using the term dorsal. Is there any other word I could have used as well?
Posterior.
Yeah, I could say posterior root or posterior root ganglion. That would be fine as well. This posterior root ganglion, notice, is a location of cell bodies? This location of cell bodies is a location, and I will tell you right now that here in this dorsal root ganglia, we have a bunch of cell bodies that have their axons come next to the cell body and then travel and come into the backside of our spinal cord. Well, based on what I've drawn there, what does that look like as the structural classification for this neuron? How many processes does it have coming off of it?
So a unipolar?
Yeah. This is a unipolar neuron. And if it's a unipolar neuron, what is its function?
Sensory neurons?
Example.
So do we know they're cell bodies just because you tell us they're cell bodies? Like how we wouldn't really.
The same way in the Purkinje cells, I showed you the low magnification view so you can see it in context. Let's take a look at it up close. I want to keep all the words, so let's get rid of the pictures. Notice here, again, we're seeing the dorsal root coming in, and then here are all the clusters like nests of eggs, all these clusters of the cell bodies. Now let's take an even closer look. Whoops, didn't want to do that. I want to keep the words. Bingo. Again, notice if I showed you this right off the bat, it wouldn't be really in context and it would be hard to understand. But now we know that we're looking at one of those nests of cells. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23, and so on and so forth. There's a couple things here that are a dead giveaway that we are sitting inside of a ganglion. The first is, notice on the outside of these cells have these smaller nuclei like crowns around their head. These are a special type of glial cells that wrap around and help and support these cells that happen to be called satellite cells because they wrap around them like satellites orbiting the Earth. So when you see those satellite cells, that's a dead giveaway that you are in a ganglion. And let me go ahead and remove all my drawings. No, not the words. I want the words. While we saw almost 24 cell bodies, how many processes do you see here?
Not a lot.
You say not a lot. Show me one.
None.
Yeah.
Because
again, I throw one apple, close my eyes and cut through it. What are the chances of me hitting the stem?
slim to none.
Right. Even if I throw 20 apples up, close my eyes and slice through them, what are the chances of me hitting the stem with 20 apples?
Still slim to none.
Yes, still slim to none. So is it surprising with 20 neurons here that only have a single process coming out of it that I'm not likely to cut through that one process?
Yeah.
So notice what we said to do in that spinal cord smear was to count the processes. We'll count the processes here. How many do you see?
None.
None. So Can there be neurons that have no processes?
No.
No. So if you don't see any processes, what must your structural classification be?
Unipolar.
Unipolar. And of course, now we have a piece of information, a solid piece of information, unipolar neuron. What is the structural class, pardon me, what is the functional classification of our unipolar neuron? Must be sensory. And so far, where's the only place that I've told you where you can find a sensory unipolar neuron? And not just any ganglion, but the dorsal root ganglion. So notice again, one piece of information gave us the other three. As soon as we figured out this is a unipolar neuron, we knew it had to be sensory. We knew it had to be a ganglion. If we saw those satellite cells and recognized that this was the dorsal root ganglion, then that would tell us that it has to be sensory and it has to be unipolar. Right? The nice thing about these three things going together is if you get one of them, you basically get the other two pieces as well.
I know this is wrong, but I thought that you can't see like the axon because of the myelin sheath or whatever. I thought that would be the reason why you couldn't see it, because it's wrapped around in it. But then...
No, and we can't, but we'll see nerves, we'll see fiber tracts, we'll see, I mean, heck, go back to the spinal cord. All of, don't use blue. All of this are axons covered with myelin. That's why this area is much more white in color. All of this area is axons covered with myelin. So we clearly can see the myelinated axons, right? We're just not seeing any off of the cell bodies in that one. Notice here, again, here we see all of the axons coming off to form that root, but we don't really see them coming off the cell bodies. We will talk, I know we haven't talked about the function of the glial cells yet. Satellite cells are not so much that they help with damage, they help with other physiological processes. So we will talk about that. But they can't so much help when the cell is damaged. Neurons can't, when their cell bodies are damaged, they pretty much are dead. Myelin, on the other hand, if you have surgery on your wrist and you lose sensation in your hand or a portion of your hand because they had to cut the nerve during that surgery, you know, in 3 months, 6 months, 2 years, that can grow back. But that's because the axon was able to grow back because of the help of that myelin sheath. But if you damage the cell body, then pretty much that cell can't recover. All right. Yes, go ahead.
So I know this is going a little bit forward, but when the cells, this is not like a regenerative tissue, right? So when the cells die, is it the microglia that like eats them?
We will get to the glial cells and we'll talk about that. So yes, I know we haven't talked about the glial cells yet, but okay. I wanted, while the neuron stuff was fresh in our mind, I know I'm adding some stuff to this, like the satellite cells here, but I promise we'll come back and do this. But I wanted to, while the different types of neurons were fresh in our mind, and I want to emphasize this relationship between not just structure and function, but structure, function, and location.
Okay, thank you.
Okay. All righty. So we've seen multipolars, both interneurons and motor. We've seen unipolar. So what does that leave us with?
Bipolar.
Bipolar. And functionally, what did we say bipolar do?
Only found in our special senses.
Excellent. And what is your most important special sense?
Eyes.
Absolutely. Yes. I will state from the beginning, again, for those of you who recall, or for those of you who don't, I am a visual neuroanatomist. I received my PhD studying the visual system, so I 100% am biased. However, that being said, 70% of the information you receive from the world around you comes in through your eyes. Over 60% of your cortex is involved in processing visual information. So just from a numbers standpoint, by far the most important sensory system in your body is your sense of sight, your vision. And here is a cross section through an eyeball. Light, of course, comes in this way through your cornea, is bent by your lens, and then is projected into this highly specialized structure located on the very back of your eye, known as the retina. And if we take an up close view at the retina, I'll stop so that you can stare in awe and wonder. This is what the retina looks like. Again, light is still coming in from here. Notice the back of the eyeball has this dark pigmented region here. This dark pigmented region is called the pigmented epithelium, and its job is to absorb light. It's dark. We are diurnal creatures. What does that mean?
I don't know.
Anyone know what diurnal means? Yeah, active, awake during the day, and active during the day, absolutely. During the day, there is more than enough ambient light, too much light to process our information. So we need this dark pigmented epithelium to be able to help us to dim the light, to increase our contrast, right? If you think about it, do you have your TV at home up to the maximal brightness so you get the most light coming off of it?
No.
No, if you do that, it kind of washes everything out and you don't see the details. And that's the same thing here. This cuts the brightness, right? However, if you happen to be a nocturnal animal, which you could be, not everybody's on camera. We could have some cats or some deers that have snuck into the class. For those cats and deers that are active during the day, during the night, they actually have a reflective layer back here called the tapetum. All right. So with this tapetum, basically it bounces the low level light back into the eye to enhance their vision at night. It's one of the reasons why your cat sees so well at night. It's also why your cat's eye lights up at night. Yes, it's also because they're possessed by demons, because all cats are, but the glowing eyes is also because of that reflection of the light. Or maybe you don't own a cat, but you happen to be driving through the woods one dark night, and a deer hopped into the middle of the road. And when your super bright headlights went into the eye of the deer and bounced off that tapetum, it actually photostuns them, where they lock into position. And so instead of running and getting out of the way of your car, you end up plowing into them. Why do you plow into them? Because of that bright light stunning them from it bouncing around. Great question. No, the red in your eye is actually from up here, not on the retina, but here in front. Oops. Here in front of the retina are blood vessels. And so that's the light bouncing off of the blood vessels when you see that. So the redness is the blood vessels, and the retina that is bouncing that light off.
So is that why we can't see that great at night then?
Yes, we do not have as good vision at night because we aren't able to amplify it the same way that the cat does.
So then the cat, like the other animals, can't really see that great during the day then? They see better at night?
They see more detail at night, yes.
Okay.
Yeah.
And then people who are blind, what happens with that? Is that
So
totally different?
there are different types of blindness. Some type of blindness does indeed start right here in the eye. Right here in the eye, all of these nuclei, and just in the circle that I've drawn, is there anybody who'd like to count how many nuclei are in there? No, but notice there's this whole big line of hundreds of millions of cells. These are the photoreceptors. They have a special segment that comes out of it. This captures the light that is come into your eye. Then out here, these cells, which are much less numerous, this one you probably wouldn't mind counting. These are the ones that are multipolar neurons. that carry this information into your brain to tell you that you've seen the light. So notice we have a third row of cells. The job of this third row of cells is to have a centrally located cell body with a single dendrite where it receives information from photoreceptors and a single axon that allows it to communicate with these ganglion cells. And lo and behold, even though you don't see the dendrites and the axons, you see the spaces where they belong. What have I just drawn?
The bipolar neuron.
Yep. So notice, as we talked about, bipolar neuron, eye, special sensory structure. And right here in this middle layer of the retina, this is where we find those specialized bipolar neurons.
And the ones to the
Bipolar,
right.
special sensory in the eye, location, structure, function. I'm sorry, what'd you say?
The ones to the right and the left of the bipolar are both multipolar.
Well, no, this is a multipolar neuron here. These
Okay.
ganglion cells are multipolar.
Oh, okay.
These are our special sensory cells.
Oh,
Remember, we talked about how there's special sensory receptors. These are the sensory receptors that allow us to perceive light. So sensory receptors, ganglion cells, and the bipolar connects the two together. And again, this is the one we care about. So if I show you this picture on the exam, I'm going to have a nice big fat arrow pointing right here. And I can ask you 3 questions. Identify the functional classification of this neuron.
sensory or special
Special sensory.
senses.
Absolutely.
Yeah.
Excellent. Identify the structural classification of this neuron. Bipolar. Excellent. Identify the location.
The eyeball.
Eye. I would accept eye. Retina is better, but I would accept eye. Eye would be fine, but retina would be definitely be better. Excellent. All right. And there you go. Locations, structures, functions for all of our types of neurons. And now we've seen them all histologically. So we talked about them here in the relationship, and now we've looked at them, histologically. Which reminds me, we got to go back. I think we have hit everything that is on your histology list now, at least for the neurons. There's one, the neuromuscular junction is on there too, I think. But if you look closely, there's one thing we missed back in the ganglion, the dorsal root ganglion. Notice as you look at these unipolar neurons, some of them are very light in color. Some of them kind of had a median color, and some of them have a very dark color. It turns out that these unipolar neurons are kind of like grandpa. Notice as grandpa has gotten older, he's starting to get those liver spots, those dark spots on his arm as he's gotten older. Do those dark spots do anything, cause any problems, have any effect, do anything? No, it's just a visual thing, something that you can notice. Well, it turns out the more active these unipolar neurons are, as they're more active, some basically build up this undigestible fat that just accumulates inside of the cell. It doesn't affect the function of the cell at all. All it does is it changes its color when we stain it. So it doesn't do anything, it doesn't have any function, but it's something we can see. And so what do you think the name of this undigestible fat that can accumulate in these unipolar cells might be?
Lipofun.
Lipofuscin.
Fusion.
Okay. Again, it's lipofuscin, it's just that, again, it's just that dark stain. So again, it's another one of those things where on the exam, notice now I could have an arrow pointing here. And I guess technically now there are 4 questions I could ask. Identify the structural classification of this neuron. Structural classification of the neuron at the point of the arrow.
Unipolar.
Excellent. Functional classification.
Sensory.
Sensory. Location.
Fine.
Dorsal.
Dorsal root.
Ganglion. Be careful, you want that dorsal root ganglion. Don't forget the ganglion part. Identify the dark substance or the dark material at the tip of the pointer.
Hypoffusion.
There you go. So I forgot to mention the lipofuscin. I thought it in my head, but then we got sidetracked by something else. And so I forgot to say that. So I wanted to come back for that. But otherwise, there you go. I think that is now it for, well, it for the nervous neurons. We still have some spinal cord stuff. We still have a couple of histology things we have to do. But those are the neurons. And that is what relates to, like I said, what we were looking at here.
I guess it would have been more helpful if I followed along with the histology handout.
Well, and that's why it's there. No worries.
Thank you.
All righty. We went a little long, but I wanted to spend the time to make sure that this made sense. So hopefully this does make sense and we can come back to the lecture now. And again, here, this is a perfect place to leave. Here is again that relationship between the structure and the functional classifications, and we cannot forget location as well. All right, let's go ahead and take our next break. Like I said, we went a little long. I apologize for that, but hopefully it wasn't too bad. We'll take a 15-minute break, so come back at 11:05, and at 11:05 we will restart, and then we'll restart, and then I will start the recording at that time. All right, so go take a biology break. Go get some more caffeine because we still have a fair amount of information to cover, or stretch, or cry, or whatever it is that you need. And then I'll see you back here in 15 minutes. Any questions before we dive back in? All righty. Okay. We've talked about the neurons, but as I mentioned, we still need to talk about the neuroglial cells. Here we see a nice picture of the central nervous system and some of the glial or neuroglial cells. Again, I'll use these terms interchangeably. That can be found here. And not surprisingly, we have different types of neurons, different types of structures in the central nervous system and peripheral nervous system. So we have different neuroglial cells found in these areas as well. As I've mentioned, close to 100 different types of neuroglial cells have been found and identified in our nervous system. We're not going to be held responsible for anywhere near that many. But what we will talk about are the 4 most common types found in the central nervous system and the 2 most common types found in the peripheral nervous system. So, let's do the central nervous system first. Starting first with the most common type, and that is what is known as an astrocyte. So astrocyte is the most common type of glial cell found in the central nervous system. When you look at the pretty picture, you can see where it gets its name. Centrally located cell body, and many processes that come off of it, giving it a star-like appearance. Astrocytes, see how simple that is? These help to provide the scaffolding, the structure for the nervous tissue. If we were in the classroom, one of the things that you would get to do near the end of this unit is do a dissection on a sheep brain. And those sheep brains, when you get a chance to feel them, they have kind of the consistency of a kind of a rubber ball. But that's because the tissue has been fixed. A brain tissue that hasn't been processed, hasn't had its proteins cross-linked, kind of has the feel, the consistency of kind of like Jell-O. So it kind of has like a Jell-O type consistency to it. So it doesn't have a tremendous amount of structure. but most of the structure that's there is caused by these astrocytes. Astrocytes are big workhorses in our nervous tissue. They are responsible for maintaining ionic concentrations, especially potassium. Remember, one of the things we've talked about is we know that that neuron potassium is going to leak out of those cation channels, just like in the muscle cell. But we want to have low amounts of potassium outside of the cell. Remember, we also talked about how at the neuromuscular junction, our neurotransmitter can diffuse away. However, do we want it diffusing away and going and finding another synapse someplace else that it can communicate to?
No.
No, so it's going to help to metabolize our neurotransmitters, help to maintain the ionic concentration, and again, it forms that structure of the nervous tissue. All three of those functions are vitally important on their own, but they're not even the most important thing that our astrocyte does. If you look closely at this picture, you will see that our astrocyte wraps a lot of its processes around these red things. Any idea what these red things might be?
Capillary.
Yeah, there you
Says it
go.
right there.
Excellent. Well, again, they don't have to be hard questions, but absolutely, yes, it wraps around the capillary. Because as we hinted at earlier, remember, as we talked about, nervous tissue does not regenerate. Or more specifically, neuron cell bodies. Do not regenerate. So our capillaries is where the exchange of material takes place. Oxygen and nutrients can come out, our neurons can get rid of waste. But do we want any old thing that happens to be floating around inside of our capillary to be able to sneak out and affect the neuron?
No.
No, we want to limit that. And so by wrapping itself around the capillaries, it helps to form an extra layer of filtering, an extra layer of protection, and actually forms what we call the blood-brain barrier. Now, is this a perfect barrier to protect the neurons? No, it is, after all, made up of its plasma membrane, so phospholipids wrapping around it. So if something was lipid soluble, like, oh, I don't know, alcohol, could alcohol sneak out through the capillary and have an effect on the neuron?
Yeah.
Yeah, absolutely. But it is fairly effective. And so it helps to limit what is able to get into and out of our nervous tissue. And if you think back to when we talked about modifying our neuromuscular junction with things like Botox or things like curare, one of the advantages that as we learn how the body works, we can use it to our advantage. How many people here have heard of the neurological disorder known as Parkinson's disease?
Yes.
Parkinson's disease, as I mentioned, is a degenerative neurological disorder. And one of the main symptoms associated, or two of the main symptoms associated with Parkinson's disease is a resting tremor. So when a person is just standing there with their hand at their side, their hand shakes. Whereas when they're making a movement, it doesn't shake nearly as much. However, the movements aren't always smooth either. They can start and stop sometimes or have incomplete movements. Again, these are symptoms associated with Parkinson's disease. And what they found is not the cause, but one of the effects of Parkinson's is a loss of a very important neurotransmitter in our brain called dopamine. Dopamine in the past year has found a lot of press. It's finding to be very important in addiction, very important in social anxieties and things along those lines. So having low dopamine levels, so things can help with that. But in Parkinson's patients, they are not capable of making dopamine anymore. And one of the effects is that resting tremor, is those starting and stopping movements. And what they found is if they can increase dopamine in their brains, most of those symptoms go away. That's awesome. Take a dopamine pill or even a dopamine injection in your arm, and suddenly no more symptoms. Well, it's not quite that easy, because as it turns out, dopamine is not able to cross the blood-brain barrier. So can you take a pill or an injection in your arm and get that dopamine into your brain?
No.
No. So for Parkinson's patients, what they used to have to do is drill a hole in your skull and inject the dopamine directly into your brain. And not surprisingly, even people with very severe Parkinson's symptoms are somewhat hesitant to have holes drilled in their head. So is that a very worthwhile way or successful way of getting dopamine into the brain? Useful
No.
way?
Isn't there something that like they have that like it creates dopamine and like it
Exactly.
absorbs or something?
Exactly.
Right.
Augustina, there are some mental illnesses where they would do things like that, not dopamine at the time, or like we talk about frontal lobotomies and things like that, and we will get to some of those things. Yeah, the lobotomies. We will talk about that when we get to the nervous anatomy. But in this case, recent studies have shown dopamine is playing a role in that. But luckily, because of these Parkinson's patients, there were some researchers at a pharmacological company that realized that the brain doesn't just make dopamine out of thin air. The same way you don't make that big, huge Disney castle out of Legos just out of nothing. You've got bits and pieces that you have to assemble and put together to make it. And as Ava pointed out, one of the bits and pieces is a chemical called L-DOPA. L-DOPA is a precursor to dopamine, which basically it means it's a chemical that gets turned into dopamine. And it turns out L-DOPA can cross the blood-brain barrier. So while you can't take a dopamine pill, if you take an L-DOPA pill or an L-DOPA injection, that can cross the blood-brain barrier, get into the nervous tissue. And once it's in the nervous tissue then turns it into dopamine. So we can get rid of those Parkinson's symptoms, and nobody has to have holes drilled in their heads. Parkinson's patients are happy, the doctors are happy, the shareholders in the pharmaceutical company are happy. Everybody's happily and lives happily ever after from this. All right, so again, it's another great example of how we learn about these things and we change the way that we handle issues as we understand how the body works. All right, so again, as I mentioned, forms that blood-brain barrier, provides structural support, metabolizes neurotransmitters, and helps to regulate potassium levels in the interstitial fluid. Questions on that? All right, let's look at our second type of glial cell. It is an ependymal cell. These are actually epithelial cells. So again, as we know, that means it lines surfaces. And in this case, it lines surfaces both on top of and inside the brain. As you know, inside the brain, there's fluid that surrounds your blade, so it's not your brain, so it's not constantly bouncing against your skull. But there are small cavities, holes inside your brain that are filled with that fluid as well. And what is that fluid that is inside and around your brain?
Cerebrospinal fluid.
There you go. Cerebrospinal, all one word. which of course, right, always gets abbreviated on, House and all those other medical shows, Grey's Anatomy, right? We got to measure their CSF. Cerebral spinal fluid is basically filtered blood plasma. But again, remember, as we talked about, do we necessarily want everything that's in the blood plasma getting into the spaces around and inside of our brain? No. Well, so luckily we have these ependymal cells, which again play an important role in helping to filter and regulate and circulate that cerebrospinal fluid. So it's going to lie in the internal and external spaces of our nervous tissue. All right. Questions on that? If you learned anything in anatomy and physiology, there is always going to be an alphabet soup term. Oh, wait, hold on. There are certain names for those spaces. They're the lateral ventricles, the third ventricle, fourth ventricle. So those spaces inside are the ventricles that are filled with cerebral spinal fluid and lined by the ependymal cells. But they're supposed to be there lining those surfaces. So I don't think that there is necessarily a special name other than, like I said, the ventricles are the names of those spaces. All right. Oh, so like I said, the brain is completely I think I know where you're going with this. The brain is completely surrounded both internally and externally by cerebral spinal fluid. So again, let's just do a real simple drawing. Here's the outside of the brain, and then here's like a couple of those spaces on the inside. So we have our cerebral spinal fluid bathing the outer surface and filling these spaces. If cerebral spinal fluid was to somehow get into the tissue, there would have to be damage in the tissue for that to occur. So the damage in the tissue would be more of a problem than the cerebral spinal fluid getting in there. What I think you might be talking about is there is, without getting too much in detail, there are some connective tissues that surround the outside of the brain and help to protect it. And in there is a subdural space. And if blood fills up, if you get some kind of a head injury, you can get blood in this space and you can get what they call a subdural hematoma. Was hematoma what you were thinking of? Yeah, so hematoma is actually blood in one of those spaces. So there can be swelling of fluid, but in this case, that fluid would be blood in that subdural hematoma. So yeah, that's related to blood. And we will talk about that when we get to the connective tissues, but it doesn't relate to the cerebral spinal fluid. Excellent. All right. There's always that alphabet soup word you're going to have to learn. And here it is here, oligodendrocytes. But luckily we already know what it does. Similar to the astrocyte, it has a centrally located cell body with multiple processes that come off of it. However, notice these processes, these extensions of its plasma membrane, notice it's wrapping around the axon hundreds of times. And what was that fancy material that we said wraps around the axons?
Phospholipids.
True. Well, it is the plasma membrane that's phospholipids, but remember we
Myelin
said it
sheath.
causes the myelin sheath. So it forms the myelin sheath. But you're right, the myelin sheath is hundreds of layers of the plasma membrane, phospholipids. And again, this is what makes our white matter. That bundle of plasma membrane wrapped around these axons, making our white matter in our central nervous system. So then the question becomes, why? Why would we want each of these axons to have hundreds of layers of fat wrapped around them?
For
Because
protection.
it makes the action potentials travel really fast.
Excellent. Perfect. You guys both hit it on the nose. I didn't see who wrote insulation, but let's see. Oh, Mitch, and then also Augustina. Absolutely. We want to protect the electrical signal. We want to protect that electrical signal, right? Just like in your wall, there are copper wires carrying electrical signal everywhere. But are they just all loosely bundled together? No, because then that electrical signal could cross from one wire to the next. So when I turned on the light switch here, instead of my light coming on, the garbage disposal turned on downstairs, or the garage door opened, right? Or the TV turned off, or any number of other things. We want to keep those signals separated from each other. However, as was also mentioned, I think Ava was going to mention it, the plasma membrane wrapping itself hundreds of times around the axon actually makes the action potential faster. It can actually be 5 or 6 times faster than if it isn't myelinated. So not surprisingly, most, not all, but most axons are going to be myelinated. It protects them and it makes them faster. Lucky for us, there isn't any kind of, I don't know, autoimmune disorder that would target these oligodendrocytes, destroying them, messing up those electrical signals as they're trying to travel throughout our body. Luckily, nothing like that happens. Or does such a thing exist? Is there a disorder that causes many small cracks to occur in this myelin as we get broken down by our autoimmune? There you go. Multiple sclerosis. Multiple sclerosis is an autoimmune disorder where our own immune cells target the oligodendrocytes, destroying your axon's abilities to carry signals from one location to another. All right. Lastly, and this gets to Ava, what you were talking about before, our fourth and final neuroglial cell in the central nervous system is known as a microglial cell. These cells are similar to our white blood cells. In that they're phagocytes. Remember, phagocytes go and find something that doesn't belong and gobble it up. So if you have an injury to your brain and some harmful bacteria or viruses get in there, these microglial cells can migrate to that area and destroy that. But if there's any nervous tissue, any neurons that are destroyed, they can go in there and clean up all those dead and damaged cells as well. So they provide that very important protection. But here's the problem. When you have an injury to your nervous tissue, do those neurons regenerate?
No.
There's no regeneration. So basically, when these microglial cells go to the area and destroy the cells and fill that region, they basically produce scar tissue. And is that scar tissue going to function the same way as the nervous tissue before?
No.
And we have a fancy name for that, a traumatic brain injury. Right?
Was there like a study done that kind of proved that sometimes these were not the best to like
Absolutely. So remember, as we talked about before, the more we understand these systems, the more we can use them to our advantage. And let's talk about that study you were mentioning. Now, I will start from the get-go to mention 3 things. Oh, is there a Netflix series on this? Okay, well, there's been, again, this is a
Or several.
there's a very,
Yeah.
very heavily studied, I didn't see the Netflix special, but what I can tell you specifically about is a study that took place at UC Davis. So it happened locally. However, I will tell you 2 things right off the bat. These studies happened many, many years ago. So please do not grab your pitchforks and your torches and your signs and go protest at Davis now. These are not occurring at Davis anymore. And I promise you, everybody involved died very slow, painful deaths. Okay, so everybody got what they deserved from this. But let's talk about this study. This study involved rats and a swimming pool. They would get a little kiddie pool, and in this kiddie pool, they would fill it with water. And inside this kiddie pool, they would put a plexiglass stand. And because that plexiglass stand was clear plexiglass, was that easy to see through the water?
No.
No. Now, rats happen to be okay swimmers. They're not the best swimmers, but they're okay swimmers, but it takes a lot of energy. So they're just randomly swimming around the pool. And as they randomly swam around the pool, they would randomly come across that submerged table. And what they could do is they could actually stand on that submerged table. Their head would be above the water. They wouldn't swim anymore. And so they would be happy. So you put the stand in there, you put the rat in the pool at some random location, and they randomly bump into the table. Then the next day you come back and you put the rat in the pool, and what does the rat do?
Swims there faster.
Yeah, it
knows
pretty
where it
much
is.
swims straight to the table. And you can time how long it takes the rat to do that. Once he has learned where that table is, he's going to pretty much swim to it right away. And that's that. Then what they would do, because this is science and everything needs to be standardized, they would take the rat and they would connect a long hollow tube to the rat's head. This long hollow tube was a standard length to which they would then insert a standard size weight so that it fell a standard distance and hit the rat on the head, inducing a traumatic brain injury. Again, all these people died horrible, slow deaths, so it's okay. They then would take this rat with a traumatic brain injury and they would put it in the pool. And guess what happened when they put it in the pool?
A longer time for it to find the stand.
Yeah, it took a longer time for it to find the table. Because they had incurred a traumatic brain injury, it decreased their function. All right, you with me so far? Excellent. Then they took a second bunch of rats. And with this second bunch of rats, they did the exact same thing. They trained them to find the table. They counted how long it took them to get to the table. They introduced a traumatic brain injury. But this time, when they introduced that traumatic brain injury, they also into the brain injected a medicine. And what this medicine actually does is it basically it retards the function of the microglial cells. So the or suppresses, let's say I like suppresses better. It suppresses the function of the microglial cells. So the microglial cells aren't able to get over there to kill any pathogens, but they aren't able to get in there and destroy the nervous tissue either. And what they found is when they put these new rats into the pool, these new rats actually did a much better job of finding the table than the ones that had just incurred a normal traumatic brain injury. Right? Again, this is a long way to go to find out this information, but basically what they discovered is that these microglial cells, while very important for helping to deal with harmful pathogens, are very aggressive. Right? They're in the, you know, kill them all and let God send them out, you know, sort them out kind of philosophy. Yes, there may be a dead neuron there, and they will happily gobble up that dead neuron to get rid of that tissue. But that damaged neuron, did they bother to wait to see whether it survived?
No, they just
Nah, it's
kill it.
probably not going to survive, so we'll just destroy that one. And that one with the sprained ankle, well, that could become infectious. We might as well kill that one too. And that one with the black eye, we'll just kill that one too. And that one just sneezed, we'll kill that one too. Turns out they were a little bit more aggressive. And if you suppress their functions, many of those neurons that were damaged are actually able to recover. They don't die. Instead, they're able to recover. And the more neurons you have recover, the more function you're able to maintain. And
So
it is,
do
go ahead.
they use this for a lot of traumatic brain injuries now?
Yeah, and if you think about, and again, probably one of the places where you see this the best is in sports. Even 10 years ago, if a football player had a, you know, spinal cord injury, right, they basically would roll them off the field to get them out of the way because they were basically going to be paralyzed for the rest of their life and we needed to get on with the football game. Now, they stabilize them, they bring out the carts, they stabilize their head, they don't take their helmets off, right? When they get into the ambulance, they put cold compression on it, their medications they give. Our whole way that we deal with traumatic brain injuries and spinal cord injuries have changed dramatically because of studies like this. Yes, it's not nice what they did to these rats. And again, I'm not condoning it. I'm not saying whether it was good or bad, but it is studies like this that have provided us insight into how our brains work. And that has helped us to understand function. And as we understand function, we can modify that. So now not only do they treat spinal cord and traumatic brain injuries very differently in sports, but athletes are much more likely to recover from them much more rapidly. Rather than pretty much being paralyzed for the rest of your life, you hear stories about, what was it, gosh, 3 years ago on opening kickoff in football, there was a guy from Buffalo who had a spinal cord injury and was completely paralyzed, and he was actually walking by the end of the season, right? So was there another CTE is a completely different so a lot of those NFL studies also involve CTE, but which are more from the, rather than major impacts, are from the constant impacting and the chronic effects that those can have. So yes, there's a lot of research going on that. And again, you see now they're changed the rules on the kickoffs, they're changing, it's penalties now when you're leading with your head and all this kind of stuff. They're both from a research standpoint and from a playing standpoint, they're changing the rules to try to protect their athletes better. So yeah, so again, these things can sound a little gruesome on paper, but and they were a little gruesome, but we have learned a tremendous amount about how the body and the brain works, and so there is some benefit to that. Again, if you want to have the philosophical discussion about whether that gain is worth the cost, that is a valid argument you can make, but you cannot argue that we gained information. You can just argue about the cost of that and whether it was worth it. All right, and like I said, I was not involved. I wasn't even at UC Davis when it was there. I am old, but I'm not that old. And like I said, all those people are gone. All of them died slow, painful deaths. So I'm sure they all got what they deserved. All right. So that is that study you were talking about, or one of many studies that was like that. But that's the one that's close to home because we're close to Davis. All right. Questions on those? All right, those are the 4 neuroglial cells you need to know in your central nervous system. Oh, I'm sorry. Go ahead, Augustine.
How does like alcohol and like drugs play like an effect on, I guess, like this section that we're talking about?
So it's really a combination of these cells and also these cells. As we mentioned, our astrocytes form that blood-brain barrier, limiting what can get past the out of the capillary into the nervous tissue. The problem is when you're dealing with something that is small, like some of the toxins that we take from chemicals can be, or more importantly, something that is lipid soluble, like alcohol, right? Alcohol is lipid soluble. It likes like. So is there really a number of phospholipid layers we could put on the outside of this that would stop alcohol from being able to diffuse out of the capillary into the nervous tissue?
Mm-mm.
No, not really. So as a result of that, things like alcohol, things like drugs can come out and affect the neurons.
So is that why they found like that L-dopa? Because I think some drugs can increase your dopamine levels, and they didn't give that to the Parkinson patients first. They didn't try that. Like, aren't there some drugs that increase dopamine levels? So wouldn't that be the easier route to go?
True. The problem that you have is many of those or some of those drugs stimulate the cells that make dopamine. And part of Parkinson's is those cells are destroyed. So if those cells aren't there, then
They can't. Okay.
you can't increase their activity. If there's no workers to
Okay.
make it, it doesn't matter what your incentive is. They're not going to
Right.
be able to make it. But if you give the precursor, then other cells are able to make it. So then they get in here, they can impair function, right? So again, obviously, when you're if you've partaken of numerous alcoholic beverages or other types of behavior altering drugs or medicines, it will affect the function of the neurons, and it also runs the risk of damaging the neurons. And so if those neurons get damaged from that chemical, then what's going to happen is those microglial cells will come destroy that damaged neuron, replace it with scar tissue. And so then you run the risk of chronic drug and alcohol abuse, majorly impacting the function of your nervous system. Yeah, soccer is another great example. You know, NFL, again, we're talking about CTE for these types of studies, but NFL gets all the highlights for that because of the money that is made and goes into the NFL. But yes, soccer players with the headers, with the collisions, for soccer players, it's even more severe. I mean, they've changed the rules dramatically. I think at most high schools now, or at least definitely at all junior high and youth levels, heading the ball is illegal. You're not allowed to do that anymore. And I think more and more high schools are starting to do away with that as well. So yeah, soccer, it's even more of an issue. And more of an issue for females as well. And again, why doesn't it get the attention that the NFL does? Yeah, again, yeah. So again, but you know, females and soccer, those don't get the attention that, you know, NFL does, and so they don't talk about it as much. But you're right, there is more and more awareness, and so it is improving. Still not where it should be, but it is improving. All right. 4 central nervous system neuroglial cells, 2 in the peripheral nervous system. And not surprisingly, they are going to serve similar functions to what we saw in the central nervous system. One of these we already talked about. Notice here is our friend, the satellite cell. Notice, as we can clearly see from this illustration, the satellite cells are glial cells that wrap around the cell body of a particular structural type of neuron. And what is this structural type of neuron that the satellite cells wrap around? Come on, what's the structural classification of the neuron you see there in front of you? Unipolar, excellent. Oops. And so what is the functional classification of those? Not the special senses. Remember, special
Sensory.
senses are bipolar. So just the sensory. Excellent. So, and remember, oh, where did we find these? Where did we find these unipolar sensory neurons with their satellite cells wrapped around them?
In the ganglia.
And how many ganglia have you been responsible for so far?
So the peripheral ganglia.
Well, but there we go. One, dorsal root ganglion. There you go. So remember when we looked at those ones, now notice with the illustration, we can see the stem, we can see the one axon coming off of it. And we can also see those nice satellite cells that wrapped around it. Remember, these basically do the same job as the astrocytes. Obviously, they're not forming the structure, but they are going to maintain the ionic balances. And they are going to metabolize neurotransmitters. And they're going to provide some protection. And those unipolar neurons found inside of our ganglia, our dorsal root ganglia. And notice also our axon has some nice fancy sheets over the top of it. In this case, they are going to be surrounded by a special type of cell called a Schwann cell. Now notice, hold on. Like we've talked about, we see how they show in the illustration. Our Schwann cell goes to the portion of the axon, wraps its phospholipid bilayer hundreds of times around that axon, forming this very dark, distinct structure around the axon called the myelin sheath. So it is obviously similar to the oligodendrocytes, the same way we saw before. However, remember an oligodendrocyte can form, in some cases, as many as dozens of pieces of myelin on multiple axons. It had those many processes like an octopus coming off of it. Whereas a single Schwann cell wrap their entire body around one small region of one axon. So notice for this illustration that we see here, there is 1, 2, 3, 4, 5, 6, 7 Schwann cells, just forming, each one forming a small piece of myelin around one axon.
Is this to make sure that they don't touch, or why?
Well, that's just for whatever reason, you know, again, remember we want myelin for two reasons. We want myelin for insulation so that the wires don't touch, but we also want to speed things up. Now notice we said most axons are myelinated. There are some unmyelinated axons. Now, like in your wall, do you have any wires that don't have insulation around them?
I don't really know.
Hopefully,
Probably
again,
not. It's a fire
hopefully
hazard.
not. Hopefully you wouldn't. And it's the same thing as here. Notice here, for instance, we have 1, 2, 3, 4, 5 or more axons that are unmyelinated. In fact, we can count exactly how much there are. 1, 2, 3, 4, 5, 6, 7, 8, 9 that are unmyelinated. But we don't want them to touch. So notice another thing that the Schwann cell can do is it can get here in the middle, and notice it's able to isolate each one of those axons so that the wires don't touch. However, has it wrapped itself hundreds of times around any of these?
No.
No. So the good thing about these unmyelinated axons is they still do not touch. Right? We don't want to cross our wires in here, but they do not speed up. They do not, let's say it that way. They don't get the speed advantage because they're not getting the myelin. So to get the myelin, we have to take one cell and wrap it all the way around just one small part of the axon. Now, I want you to notice one more thing. Do we cover the entire axon with myelin?
No, there's nodes.
There's
like
little
gaps.
spaces in between, little gaps in between. In fact, I got another nice pretty picture here. Here it is, where we can see here is Schwann cell 1. Here is Schwann cell 2. And indeed, there is a little gap in between. And that gap, as you mentioned, is what is known as the node. And you are perfectly acceptable to call it just simply the node. However, this node happened to be found by a Frenchman, good old Bob Ranvier. And Ranvier is incredibly fun to say. So if you want to say node of Ranvier and spell node of Ranvier in the French, then you are welcome to do that. But you're also welcome to just call it the node. But like I said, Ranvier is very fun to say, and it's very French. And again, here we see the difference between unmyelinated axons that don't touch and a myelinated axon where that Schwann cell or that oligodendrocyte wraps its plasma membrane hundreds of times around this cell, providing that insulation. And those are the 6 glial cells you're responsible for. Questions on that? All right, how are we doing on time? All righty. Let's see where we're at right now. Oops, not what I wanted. Hold on. All right, this is a good stopping point for the lecture. We'll stop here for the lecture. Oops, what did I just do? I don't know what I just did. But what I do want to continue to do now with a little bit of time that is left is I want to talk about our brain anatomy. As I mentioned, that brain anatomy handout, and let's find that handout. I don't have it there. So it's in the modules. As I mentioned, this gross anatomy of the nervous system and the histology is probably going to be about 50% of your lab exam. And as you've looked at this handout, there is a lot of material. So notice when we had a lot of material with the bones, we divided it up: axial and appendicular skeleton, or skull and upper limb. Or for the muscles, we divided it into muscles that move the head or muscles that move the leg, right? We were able to put it into more easily digestible pieces. And when we took smaller chunks of it, it was easier to learn that information. And we are going to do the exact same thing with the nervous tissue. Your nervous tissue starts developing very, very early, within the first couple weeks of your development, as you're becoming that fertilized egg and becoming that zygote and becoming that embryo and that fetus. And what happens is you start out with just a single layer of nervous cells that then gets a little bit of a fold in it, and then it rolls up into a tube. And that's what we see here. Notice this pretty little illustration shows us a tube that forms at about 3 weeks of age. Then by 6 weeks of development, that tube has started to get some specialization. And notice as they've color coded it for us here, not 1, not 2, not 3, not 4, but 5 different enlargements. What your book refers to as the brain vesicles, but what I think is more appropriately referred to as the cephalons. And notice these cephalons have fancy names: telencephalon, diencephalon, mesencephalon, metencephalon, and myelencephalon. And the good news is, while you have 3 M's, I will note that, as you can see there in alphabetical order, mes, and then met, and then my. So that helps to keep them what they are. And even though you're pretty much just a bag of cells at 6 weeks of development, these 5 cephalons, these 5 enlargements, become specific parts of your brain. Notice the part that is the telencephalon becomes the largest region of our brain, the cerebrum. The area that's the diencephalon actually stays called the diencephalon, as is the area that's the mesencephalon, although sometimes for the mesencephalon, you will also see the term midbrain. And so we will use those 2 terms interchangeably. We can say midbrain or mesencephalon. Our metencephalon becomes our 2 enlargements. One, the cerebellum, which we already looked at. That's where we saw those Purkinje cells, and another enlargement called the pons. And the myencephalon becomes all of our water boy fans, the medulla oblongata. And here we see them in name, but if you notice the colors, they correspond to the specific regions in our mature brain. So remember, we talked about that big, huge midden. That is the cerebrum. The cerebrum is comprised of the telencephalon. The telencephalon becomes the cerebrum. The cerebrum came from the telencephalon. Deep inside here, we have the diencephalon, and then the mesencephalon, the 2 enlargements that make the metencephalon, and then the myencephalon. And if we peel or cut it in half with the sagittal section, we see the same thing. Here is our, actually, hold on, let me be more specific. Here is our telencephalon. Then this kind of duck-faced region is our diencephalon. The little narrow part right in here is the mesencephalon, or the midbrain. As I mentioned, the two enlargements, the pons and the cerebellum, are the metencephalon. And then finally, the part that connects to the spinal cord, the base of our brain, is the medulla oblongata, which is made up of the diencephalon. Here, we peel the cerebellum off and we see the diencephalon, mesencephalon. Notice they removed our cerebellum, but the pons and the cerebellum and the medulla oblongata. And these things, after you peel off the two middens, we also sometimes refer to this as the brain stem. Brain stem is responsible for most of the basic life functions, things that need to keep you alive. So not surprisingly, the brain stems are pretty similar between us and a cat and a dog and a pig and a monkey and a turtle and everything else. However, where you see the biggest differences between species is in that cerebellum. That's where we do all the higher order thinking and processing. And so not surprisingly, ours is second largest based on our body size to any species. What did we say was the largest, has the largest cerebrum in relation to their size? Remember, we talked about how the whale has the biggest brain, but they also have the biggest body. So when we care brain size versus body size, we get the silver. There you go. Dolphins, remember, have the highest ratio of brain size to body size. And so why again, don't they rule the world?
They don't have opposable thumbs.
Yeah, exactly. No opposable thumbs. Screw those guys, right? They don't have an opposable thumb. We can kick their butts. All right, excellent. So you will notice, oh, we'll do that in a second. When we look at our handout, that's how I've divided these things up. First, the telencephalon or telencephalic features and all of the lists of structures you're going to be responsible for. Then we have the diencephalic features and all the stuff there you're going to be responsible for. Mes and the few things there, met and the few things there, my and the few things there. So the same way we've divided things up in the other parts of the system, we're doing it here as well. Now, notice when we look at this list, clearly the most of the stuff is here in the cerebrum. And so luckily, this cerebrum can be divided into 4.5 lobes. And what's convenient for us is our first lobe is this one here, right under your forehead bone, or in my case, the 5-head bone. And what bone is that?
Frontal bone.
So guess what we call this lobe?
Frontal lobe.
There you go. Down here on the side where the temples of my glasses are is a bone where my ear hole is connected. What bone is that down here deep on the side? There you go, temporal bone. Guess what we call this lobe?
Temporal lobe.
There you go. Notice above the temporal bone are these two bones here on the side. What bones were these?
Parietal.
Parietal. And so this is the parietal lobe, and the back.
Occipital.
There you go. So just that simply, that information we've learned once, we get to use again.
I have a question.
I don't care. No, I'm just kidding. Hold on. Let me finish this thought and then I'll answer your question.
Okay.
So again, if you look at the handout, notice under the telencephalon, we have the frontal lobe, parietal lobe, occipital lobe, and temporal lobe. So we have structures you're responsible for there. Now you'll answer your question. I was kidding before. What's up?
So when we're studying these, are we going to be tested on an actual like brain? I know we have some cadaver pictures in our like atlas, but is it going to be a mix of like both charts or illustration?
Like I said, one of the challenges of this is, for instance, the part of the brain that moves your foot doesn't look like a foot. So it can be very challenging. There are some characteristics that are fairly distinct on all brains that are on your list. Like notice, for instance, one of the things you're responsible for is a structure that is known as the central sulcus. We know sulcuses are indentations. And so this one happens to start at the top of the brain and work its way down towards our thumb. Notice if we were really careful and came back to this picture, if we really spent some time with it, we might be able to find it. Come to the top of the brain, and lo and behold, okay, that looks like that might be the right one, but it could be a little tricky, right? However, if I have a picture that happens to be color-coded like this, is a lot easier to see the central sulcus.
Yes.
I mean, yes, it's labeled, but even if the label wasn't there, the fact that it's in between these two highlighted regions, does that make it more obvious?
Yeah.
Yeah.
And how many of you actually got a chance to go on campus to look at the muscles? Did anybody get a chance to go on campus and look at the muscles in the open lab? None of you. Interesting. Okay. Anybody go to the Zoom and watch it that way? I am asking these questions out loud, right?
No, sorry.
No.
Okay. Well, remember, you do get 15 points for extra credit for going to the open labs in person or online. So there's still time left to do that. Okay. If you did, one of the nice things about the models is you're sitting there and studying with someone else, you can be like, what do you think this muscle here is? And they're all, which one are you pointing at? And the nice things about models is they have numbers on it. So you could say that one right there, number 14. What muscle is number 14? Now, again, if you looked on your actual body, would there be a number 14 on that particular muscle?
No.
No, of course not. But do having those numbers help you to study that material?
Yeah.
Yeah. Well, it turns out in ancient times, there was a neuroanatomist by the name of Corbidian Brodmann. Right now, obviously, with a cool name like Corbidian, you can see why he wasn't a Bob. He's the only non-Bob in anatomy ever. But good old Corbidian Brodmann studied brains in the late 1800s. And in the late 1800s, could they put electrodes in there and see what the brain was doing, or put them in a MRI and see what the brain was doing, or any of those kind of things?
No.
No. So what Corbidian Brodmann did is he would look at a brain of a cat. And then he would look at a second brain of the cat. And then he would look at a third brain of the cat. And what he started to notice is that, you know, all of these brains that I'm seeing have this ridge that goes down here. And then they have a groove that goes down here, and then a raised ridge sitting right next to it. Every single cat brain he looked at had this feature, this raised ridge next to this indentation that started at the top. Did he know what it did or anything like that? No, but it was a characteristic that all of them shared. So you know what he called it?
Post-central gyrus.
Nope, he called it area 1. And then he found a second area that all of these things had in common. And guess what he called them?
Area 2.
And 3 and 4 and 17 and 18 and 43 and so on and so forth. And so he gave all these areas numbers. Didn't know what they did. Didn't know if they had any specific function, but it was a characteristic that was continuously found on all of these brains. And the reason we're still talking about it is because, as it turns out, some of these distinct structures that we see from the brain have specific functions. It's not 100%, but some of them do. Area 17 is that primary visual cortex where we first see our light, first perceived light. Over here, areas 44 and 45 are Broca's area. This is where we produce speech. And so some of these characteristics, so again, do you have to memorize these numbers? No. But will we use them a little bit while we're talking about it the same way you would use the numbers on a model as we're learning them? Yes. So I'm not going to ask you on the exam, what does area 17 do? That's not going to be a question on the exam. But could I use this picture and have an area pointing to area 17 and ask you what this area of the brain does? Yes, absolutely. So will there be some images of real brains? Will there be some images of real skulls and stuff like that? Yes, but the advantage of having this online is we can use color-coded and even numbered images like this to make it easier. Because again, the goal isn't for you, what I want you to do is not spend 5 minutes on your lab exam trying to find the central sulcus, right? I don't want you to have to try to figure out what the central sulcus is on this. Is it possible? Yes, but that finding it is a waste of time. If I can give you something like this that's color-coded, then it's more important for me that you are able to identify what that is or what this area back here does or things like that. So yes, a lot of the images will be illustrations. A lot of them will be either color-coded models or color-coded images, and even some with some numbers on it.
So you can ask us a few questions, one of which, like, what is this feature? And then the second is, like, what function would this part of the
Close.
brain have?
Absolutely. So we want to be careful. On this list, for instance, these first few things on this list are simply just structures.
Okay.
A structure is a structure. A wall is a wall, right? A desk is a desk. It's just a structure. Can a desk have functions? Sure. Can there be functions to these? Yeah, sort of. But I mean, really, they're just structures that you need to identify. However, for down here, for instance, and again, I'll leave, actually, I guess I can move it up a little bit. Move it up there. And then I move this. So these are all structures, and this is how I would ask this on the exam. However, these things right here are part of the gray matter. Or what I would say is it's a specific region or a specific area. These specific regions or areas, you need to be able to recognize them. So if I point and say, what is this area? You need to do this, but you also need to know what their function is. And like I said, some of them, it should be pretty obvious. What do you think the function of the primary visual cortex is?
Vision.
Yeah. What do you think the function of the speech area is?
Speech.
There you go. So again, some of them will be a little bit more straightforward. But yes, for these areas or regions, if it's an area or a region, you need to know their function, right? The central sulcus doesn't have a function, but it's a very distinct characteristic, a very distinct structure that we can learn. And notice one other thing. Notice on our cerebellum, we had folium were the raised ridges, and the sulcus is the indentation. But on the cerebrum, it's different. We still have raised ridges, we still have grooves, and the groove is still a sulcus, but now the ridge is called a gyrus. And of course, the plural of that is gyri. Oops. And because anatomists love to name things, a sulcus is a groove, but sometimes you can have a really deep groove. Any idea what we would call a really deep groove? A really deep groove is a fissure. So guess what? The longitudinal fissure, oops, whoa, is a really deep groove that runs longitudinally, separating the brain into two hemispheres. And notice also, now that we have some anatomy, guess where the pre-central and post-central gyri are located? Where's the pre-central gyrus located?
Before the central sulcus.
Yeah. And so then where would the post-central gyrus be?
After.
Yeah, exactly. So notice if we go back to the pretty picture. Oops. There we go. Notice again, they've color coded it for us. Here in red, in front of the central sulcus is the pre-central gyrus. And here in dark blue behind it is the post-central. And notice there's also color coded it by lobe for us as well. We have our frontal lobe. We have our parietal lobe. We have our temporal lobe, and we have our occipital lobe. So again, sometimes these color-coded images can make it a lot easier to understand. All right, we will go through the anatomy list like we've gone through most of our anatomy lists together, talk about identifying these things, talking about their functions, but it is something that you can start looking at, and we'll start doing that on Wednesday. And then also now that we've talked about the anatomy, we'll start talking physiology of the nervous tissue as well. All right. Questions on any of that? All right, so we've got the anatomy in our books for the nervous tissue. So now we can talk about how the nervous tissue functions in the next class, and then we'll start working on this brain list that we've started as well. All right, that's all I've got for you today. We get to finish a little bit early unless there's any questions. All right, I will be in office hours and I will be working on grading. So I'll try to get your exams done as soon as possible. Like I said, as soon as they're graded, I will get the grades posted, and then the exams will be posted after class on Wednesday as always. All right, you guys have a great day, and I will see you on Wednesday.